Well, good evening, or good afternoon, or good morning, wherever you happen to be. We have just dropped in to 2,000 meters deep on a mountain, an undersea mountain on the Kane Fracture Zone near the intersection with the Mid-Atlantic Ridge. And we're doing a dive to check out the geology and look for signs of hydrothermal activity. This is the second dive of this expedition. We had one dive a couple 100 kilometers south of here, and we're going to be working at this area now for probably up to a week. It's a very large area to cover, and we've got two AUVs, autonomous vehicles, in the water right now, mapping out about 20 square kilometers of the seafloor in a slightly different location on this undersea mountain. And we'll be looking at that data when that comes up tomorrow. We've managed to fit in an ROV dive before the recovery of that, so it gives us about 8 hours or so of time on the bottom. So we'll be ending this at about 5 o'clock in the morning our time, which is 3 hours earlier than universal time, 5 hours different from the west coast of the US. So we are looking at a sedimented slope with some rocks exposed, and we're about 2,000 meters, and we're going to be slowly climbing up the hill from here, making observations as we go, looking at the rocks, taking some rock samples, seeing what animals are living here on some of the rocks, corals, sponges, other deep sea fauna, and looking for signs of that reaction between seawater and mantle rocks if we happen to find exposures of those. Sure, I think that's a good way to start. Yeah. Okay.
That's not too big.
So I've got, I'm Dave Butterfield, by the way, and from the University of Washington and NOAA Pacific Marine Environmental Lab, and we're out here on the Schmidt Ocean Institute vessel Falkor 2. It's the first science mission for this vessel, and we're really impressed with the capabilities that the ship has and the amazing crew and support team out here. So we may start this dive by picking up one of these rocks and taking a sample and getting a closer look to see what type of rock it is. Most likely basaltic lava, not sure how old, whether it's in place as a lava flow or pieces that have fallen down the slope. But in a second here we'll move in and try to find a rock to sample. So why don't we do that, Adam? Oh, sorry, Joe. Adam was there a minute ago.
So you want to land out here and get a sample possibly?
Yeah, let's go in there. And you want to try one of those in the back? That's try one of those on along that line there in the back.
Or on the right side could be easy.
When you say the right side, what, over in here? Okay. Okay. So she's saying maybe center left of your screen.
Yep.
Breathe in there. When do you want to turn on the?
Right away.
Okay.
Turn it on. Oh, you've got your pump control, right?
I have the pump controller here. Yeah. I think they have to. I don't know if they have to turn on the.
This is on the fluid sampler is on, and turning on the flush pump, so you've got water coming through there. And give that a minute, and anytime you want. Take samples. You need your power.
And also maybe for the
Could we get the power on to the methane spectrometer? Okay, we're just getting settled in to start this dive. There's a lot of various things going on. We've got a hydrothermal fluid sampler on that pumps water from vents that we find through a long set of Teflon and titanium tubing. And we've got various other things connected to that. One of them is a new methane spectrometer that uses a laser absorption in a capillary. We pull seawater, small amount of seawater into a pressure case with the instrument and extract the gas out of it in a vacuum, and that gets circulated into a capillary tube in a laser, and we measure light absorption through that and come up with the concentration of methane in the water. So Kaori is our methane spectrometer engineer out here, and she's operating that behind us. And I've got the fluid sampler here in front of me, the laptop computer controlling that. So now I'm going to talk about rock sampling here. So, okay. There are some small pieces. You just want to, when you're ready to get in there, see if you can break a piece off or pick up one of those small pieces that's next to these larger rocks.
Okay. Yeah, Ben, so he's saying right in here, take a look at these ones, see if there's any way to break off. If it's too big, see what you get. Maybe that one just kind of lower it.
That one
Bigger than it looks. So
okay, yeah. That's a decent sized piece right there. Yeah. Okay. Why don't we get that? We'll see how it looks. Looks like it broke. Might be kind of crumbly. So you're looking at the, oh, that was a very fragile piece. That's not a typical piece of lava. This titanium arm with titanium jaws there, it's extremely powerful, but it's set up with these rubber fingers in there that sort of fold, taking a close-up view of this rock. You want to see the other side? Is there some alteration on it? It's super altered. It's falling apart. Okay, you want to keep that? Okay. Now we got options of where to put it. All right. So we're looking at a highly altered and probably old piece of lava, probably basaltic. Basalt is a composition that's typical of mid-ocean ridge rocks, produced by melting the mantle as it upwells underneath and decompresses. Most of the ocean crust surface is made out of volcanic rock of basaltic composition. There are some exceptions, but that's the dominant one.
going for it.
Okay, rock sample number 1 going in A. So whenever we take a sample, we're logging all the information about it, getting pictures of this rock that we're putting away in the basket, so that when we get those rocks back up here, we have a good idea what shape they are and what they look like, and what their characteristics are, so we don't get them mixed up. All right, that one's coming from 1,990 meters deep.
Anything else at this side?
No. Are you, you're just preparing to start a methane sample, or?
I'm just doing the weird like thing where it's like giving a bunch of symbols, but I'd probably just try to use it with the symbols, I guess, and see if it's working.
Okay.
Yeah.
You're out there, go ahead. Is that where you want it? I want to type that in a little bit further.
That was almost totally altered to,
Yeah,
I mean,
it's
to
like
clay or something.
yeah, and I think it's all manganese oxide.
Yeah.
And you can see all the light colors around it. There's quite a lot of sheep. Look at that. Like, that's all manganese oxide.
Biology team, what do you have to say about that?
That's a soft coral. Reason for guessing the idea that the Rastus doesn't. It could be other geniuses as well, but there's also
Okay, what was the possible genus?
Anthemostis.
Athamastus? Anthemastus. Okay. Okay, we're looking at a soft coral. I will probably butcher this genus name, but Anthemastus, I believe, is what the biology team is saying. Oh, do we have our lasers on? Let's turn them on. They've been on? We used two green lasers that are spaced 10 centimeters apart in parallel, and use those for measuring things that we're looking at with the camera. Okay. So while we're sitting here, I'm going to collect a water sample in the hydrothermal fluid sampler. I am going to do an HFS sample mostly to measure the methane and hydrogen concentration in the water. Those two dissolved gases tell us about whether there are hydrothermal fluids leaking out of the seafloor, which is something that we're looking for in this area. We don't really see any signs of that here, but I'm taking a sample, whether it's just a background sample, or whether we'll find that there's some trace of hydrothermal fluid leaking out here. So the time. Okay, so I've got HFS number 17, bag 17 coming up. Sample 2, right, okay.
Can you go back and zoom on that rock, but not all the way in? I just want to look at the light.
I'm just going to say.
Okay, this one here.
Yeah, just zoom in the prior. Hold it right there.
Okay.
Are you logging? Are you logging?
Yeah,
Okay. Okay, so ready to start now. Starting sample. We have a logging system here that anyone with a Wi-Fi connection to the logging system can log samples. Got Cécile Catalot from France, from Ifremer. She's another vent fluid chemist, and she's logging the information about this sample. We can see, where's that picture of the rotary flow meter on the front?
Turn on SEHD1.
So it just turned past midnight universal time. There's a fish. So it's now March 15th, universal time for recording our samples. On this sample? Oh, wow. Well, we've seen a fish and a shrimp already, and there's this, look at scale worm there. Scale worm coming out from underneath. There's another shrimp flying by, just off the side. This rock also is looking extremely altered. Like if we touched it, it would probably fall apart. Seeing what? Oh, yeah. Scale worm is crawling up out of that crevice where the shrimp was hiding. These are they're predators, those worms. There's our shrimp. Okay, this sample is almost done. Okay, that sample finished, and we got 450 milliliters. Temperature is ambient, 3.5.
Maximum,
Yeah, 3.5. Well, the average says 3.4. There was a point. I guess it. So the deep water's pretty cold. Okay. Okay, well, we should probably start I think I'll start another long sample, which is a filter for DNA. And hey, Bailey, I'm going to start your sample, I think. And you guys have 13, 14, 15? I'll just do that. Okay. Okay. We could, sure, we'll do number 12. We can move when we do this, so. Okay. No, I don't need that. Ground fault on the camera? Okay. Okay. Yeah, I don't need to see that. Okay. Yes.
That's a good question for Monica. Yeah, so
What's all the debris? That's.
okay, so we're saying it's probably the mob material, whether that's the shells, pteropod shells, spicules from sponges, gastropod shells. There's a lot of mixtures of things.
It's some combination of, biological debris, shells, spicules, stalks. There's an awful lot of it right there, and I'm not sure why. This is pretty old sediment in here, so it's had a long time to accumulate. There's our lasers. Some sponges, coral. Could one of you biologists come up here and talk? Come on. Come on, Monica. Monica, come on. I want to hear you talk about what we're looking at. I'm going to do a filter for, so it's going to be a long one, so they just want a background filters.
Okay, so we're seeing a lot of fragments from shells, so pteropod shells, possibly sponge spicules, gastropod shells. The sand is full of a lot of biological debris. We're seeing lots of sponges, possibly tunicates, but I haven't confirmed that. It looks like corals, but they could also be hydrocorals, so we're not exactly sure what we're seeing here. We saw a scale worm earlier and a shrimp, and we're seeing some empty tube
Is that
tubes
black?
and
Is
shells.
that black thing a tube?
Yeah,
in the
that
back
looks
there?
good.
Yeah. Zoom in on that.
We also have a crab here on the right coming out, or at least cod lobster type.
Oh yeah.
Yeah.
Those long pinchers.
Yeah. Sean, can you take a screen capture of it? Thanks.
These animals seem happy living in little crevices of this altered rock.
There's also what looked to be another, possibly bamboo coral.
Yeah.
Small one.
So the black tube there. Can we zoom in on that?
Yeah.
That's it. Okay. Maximum zoom. All right.
Oh, lasers, right.
The lasers are on.
Lasers are
They're
on,
just
but-
not in the camera view.
Where does this? Yeah. Okay. It's hard to tell.
Right there is the laser.
Yeah, gotcha.
So if you want to measure, just let me know and I'll zoom out. Come on, whatever you want.
Yeah, so I think the coral that we saw was a little bit more over this way, I think, but yeah, that one, that little white guy on the rock. Okay, so that looks like a hydrocoral to me. Yeah, so a hydrocoral looks a little bit different because each of those little balls would have its own like feeding apparatus, like polyp almost, but it's not a true coral. It's almost like a hydroid.
Okay.
So that looks like it to me, but I am by no means an expert. So
Can you define what a hydroid is for people?
it's also a cnidarian, but it's not a coral. So I don't know how to Sean, is it a cousin of the coral? A cousin of the coral, sure. Yes, say that. Just how to define a hydroid. Cousin of the coral.
In the same family,
Okay.
in the same family or whatever it's called.
Yeah.
There's a lot of biological debris out here, as well as a lot of live animals here.
I'm also wondering if that right above your lasers right now, if that's a sponge or if that's just like what is causing that. Yeah, that's crazy.
There's some shells, little tiny shells.
Yeah, and so it looks like it looks almost like glass fibers, and that's what I think are sponge spicules. So sponges are made out of like silica, basically like needle-like structures called spicules, and they make up a skeleton, a really intricate skeleton of a sponge. And when they die, you can pouch things like this, where like sediment can accumulate and form these interesting structures. And I don't think this is a live sponge, but again,
It doesn't look very alive.
no, it doesn't.
Yeah.
They almost look like some of the
But-
debris are broken shells from whatever.
Yeah, this is really
Yeah,
interesting.
there's lots of stuff here. So evidence for
Yeah.
a lot of life living here for a long time,
So there has been
and some very old rocks.
Do you see the thread? Or
We
something? It's under the rock in the middle on the bottom right.
oh, yeah.
Yeah.
bottom right.
Poking out.
Oh yeah, there's a crab poking out there too. We saw a similar one up above. Yeah. Oh, he's looking right at us. Wondering what that bright light is. Huh. Yeah, these animals live in total darkness, except for the bioluminescence. Okay, are we ready to start our transect up the hill?
Do you,
We're going to be facing east, going up.
do you consider changing routes if you find anything along the way that makes you deviate?
If it was something really striking, yeah.
Okay, I was just wondering.
But we're just going to go pretty much straight up the hill, right towards the summit, to the very peak.
Okay.
Yeah. There was a
Directly towards waypoint 2, I guess.
yeah. Do you have waypoint 2 in your system? Heading to it, it should be pretty much east.
Yeah, I've got a line there to track.
Okay. East and a little bit north.
Yeah.
So we can go at a pretty quick pace too.
Okay.
Okay, starting at 1,990 meters, we're going to go up to close to 1,000 meters at the very summit. So about a 1,000 meter rise.
ROV bridge.
What would you say the slope is in some of this?
This is pretty gentle so far.
But I mean the average that we're going to see over this,
Do you
like
know the average
30%
slope?
or something?
Just like is it 45 degrees?
45. Okay. So average slope for this feature that we're going up is going to be about 45 degrees. And we're going up a long scarp that's probably 10 kilometers long. And at the top is the highest point of this massif on the inside corner of the intersection of the Kane Fracture Zone and the Mid-Atlantic Ridge. It's an oceanic core complex where the volcanic layer in some places has been stripped off through what's called detachment faulting. The mantle comes up and is exposed at the seafloor in some places. However, what we're looking at here is sort of a vertical section of that, and on top will be probably volcanic rocks. So we're looking at a section where the volcanic layer is still there. There are other portions nearby where the rocks exposed are normally buried by the volcanic layer, but here on the oceanic core complex, they're exposed at the surface of on the seafloor. And we're looking for hydrothermal activity that is created when seawater reacts with mantle rocks in a series of reactions called serpentinization. and those produce several products, including hydrogen through reduction of the water and oxidation of iron in the minerals from the mantle, olivine being one of the primary minerals that gets altered. All right, here's some, looks like talus or loose rocks that have tumbled down the slope. Possibly a cross section of a pillow lava there, broken pillow. Some of these are, there's a nice blocky one. Got geologist Melissa Anderson from University of Toronto sitting right behind me. She is going to be busy logging all of her observations about what she's seen about the rocks and the stratigraphy along this dive. And graduate student Jonathan, whose last name I'm still not quite sure of how to pronounce. How do you pronounce your last name? Umsa. Okay. I never asked you before, so. Jonathan Umsar. That's probably still not quite right, but anyway, he's also logging and keeping track of the stratigraphy that we're going through. And while we're moving, we're collecting a filter for DNA, mostly as a background sample to compare to things that we're going to take later on. For this dive, we're mostly interested in sampling the bottom water as we go along. We don't see indications of hydrothermal activity, but if you measure those gases, dissolved gases, hydrogen and methane, it'll tell us whether there's any nearby activity or and help us maybe home in on that. Sedimented section here with some what were loose rocks. Yeah, let's zoom in on that. When we come across live animals on the seafloor, we're trying to zoom in on those and get a good image of them for the biology team who are logging everything that they see. And we'll compare what we see here to animals that are found in other places in the deep sea. A lot of us are primarily interested in hydrothermal systems or hot springs, but there's also an interest in the general deep sea biology and of course the geology of this area.
We don't need to hang out too long looking at stuff.
Okay.
I just updated your position.
Copy that.
Yeah, that was about right in terms of hold up to look at them, get go in and get a good zoom on them, and if we don't hear requests from the biology team in the back to stop and do more, we'll do like 30 seconds of video and keep going.
Okay, sounds good.
This is a sign from, that's the situation. Let's walk down.
Still pumping on our background DNA sample.
Do we have the vision on the wheel?
We turned the camera off.
Okay, but it was fine.
It was going, yeah. There's a ground fault on that camera or something, so we'll keep it turned off. I'm sure you can tell me when you want another rock. How often do you want to sample?
I want things that look like they're pretty close to being in situ.
Okay.
If possible, I'm less interested in the callus unless it looks really young or has just like clearly upcoming or something. But we want to not sample too much. And who knows where this one goes from.
Yeah.
There could be some in situ stuff there though.
Underneath, yeah.
So if it looks like there's something in situ that you can grab a hold of, then that would be cool.
So wow, there's an actual star on there, huh? So looking at these rocks, you can see that a lot of them have just tumbled down the slope. Possibly there's some in places underneath. What, you want to sample?
Yeah, something in situ around here.
It looks like it's very altered stuff underneath.
I think the lack of flows just provided the heat and that alteration, but there's clearly some in situ flows beneath all of this catalyze.
Yeah, and that's what I mean, that stuff underneath looks like what we saw at the start.
This looks like the manganese oxide crust maybe on top that we sampled, which we probably didn't actually get any level with that.
You want to try break
No,
a piece? No? Keep going? Okay.
I don't think it's that difficult.
Yeah. So, for rock sampling, we like to sample rocks that are part of the formation when it was formed, not rocks that have fallen down the slope from somewhere further up.
This we could maybe grab. Oh, this looks like some flows.
Yeah, that looks like it's
That looks like a
intact.
situ flow.
But do you want it? Or you want some?
Yes.
Okay.
I always want it.
Let's try and pick up a piece there and see what it looks like. The way these rocks are laying like that, they look like they're in situ rather than something that's fallen down the slope. So we're going to see about breaking off a piece. Could be also highly altered, like the rocks below were 50 meters higher up than we were at the start. We do have a long ways to go, and we've got, let's see, well, we got 7.5 hours, so should be able to go pretty far.
I just don't think we're gonna see too much in situ. Yeah.
We're close to behind.
Okay. Okay, looks like that one's done.
We want to get a sample.
That rectangle piece in the middle.
Where?
Oh no, that one that you just kind of zoomed on, that round one there is fine.
Yeah, that looked like it might be free-ish.
Yeah.
See that down?
I'll go back there. I'll be back there.
Okay, okay. All right, thanks.
Keep walking in, I'll zoom in on it. Right there in the center, that
Yeah.
2 has 03.
Oh, got it. Yeah. So just for now, every once in a while I will take
And
sure. But for right now, I'm gonna put it back up here. They're okay with small. That's barometer. So without that, we'll give you set as well. So you can see it. And I'll port that over there. So every once in a while, I'll just look up and see it. But sounds good. And you guys want this back at HTU or certain? Yes. Well, it looks like it's gonna come through.
Yeah.
so Bailey, that sample was taken over about a 50 meter depth interval, 1990 to 1940.
Like 3 meters.
3
A
liters.
little bit of hope left in your time.
Yeah. You want to do one right now?
Survive with it. We might actually need some volcanic stuff.
Okay.
Okay, picking up a rock sample.
Can you rotate it?
Okay.
To the other side? Just all the way around, maybe?
Okay. Oh, wait a minute.
Hold it there.
Looks like we got some iron oxide surface on there and a bunch of sediment on the outside.
You guys ready to snow this?
Yep.
Okay. Watch out.
Put that one in B, I guess, next to the right of the other one. B should be fine. Yeah. Okay.
Perfect. So that means we can rock box 2. And we don't need to pick up another sample for a while. Unless
Okay.
it looks like it's like a dike or like something that doesn't have as much filtration.
Okay. So you're recording every sample on that? I think it's right there. Did you get the filter? Okay. Of course you did. We're going to take another filter too shortly. Okay, those were 2 very easy rock samples. They can often be difficult to pick up or break off if you have to break one off. Some of these are quite breakable. Fresh lavas are another story. Okay, we'll continue moving upward. Again, what we're doing out here is just today sort of getting a feeling for the geology and the structure out here. Overall, we're trying to find sources of hydrothermal venting that result from serpentinization on these oceanic core complexes. The area involved is really big, several hundred square kilometers for this massif. We can't look at the whole thing, but we've chosen a section that's about 150 square kilometers that we're going to try to map at meter scale resolution with the autonomous vehicles. And we'll also get the, here's a lava flow pointed down this slope, still intact. Nice sheet flow here. And rocks coming down the hill, when they hit this, they just keep on rolling. What angle would you suppose this flow came out at? I mean, pretty level and has been tilted up a lot? Or
No, I think it's pretty low.
you think it did flow down this hill? Like the way the shape looks? It does look like it.
In situ, like this.
Yeah.
Bridge. I just updated your position.
Transitioning into some more talus or loose rock. Let's come down just a short section of that and back into this sheet-like lava flow. Cracked and broken, but it's flowed out in a sheet that looks like it's 20 or 30 centimeters thick or so. We may eventually, depending on what's actually exposed up here, we may come to what the source of this sheet flow was, or at least we'll know when it stops. The source fissure potentially might be buried. Oh, there's a nice sponge. Wow, that is pretty.
So Dave, that was a really nice view of those individual spicules that we were seeing.
Do
possibly
you
in the
need
cemetery.
do you need to zoom in on
Nope,
that?
nope, we're good.
No,
I just
you're
wanted
okay.
to let you know.
Okay, tell me if you need to stop and zoom. Okay. Okay. Flow is broken up, and now we're back onto the sheet flow. Monica says that sponge that we just saw is potentially the type of sponge that produced all the spicules, the needle-like glassy things that we were seeing at the very beginning of this dive at, about 100 meters deeper than we are right now. These sponges and yes, I can start the filter now. Yes. Cecile, we're just gonna do another filter. You can probably log it from there. I can just tell you, but save you the squeezing into the space there. Well, come on if you want. So I'm going to start. Already selected. Okay, we're going to start filter 13. They wanted to have them close together. So I'm just starting it now. There's a fish just sitting on the bottom there. Is it what? You want to see? Either. So is this HFS filter sample 5? Okay. More lava flow, more sheet flow. This sheet flow seems to have flowed a pretty long ways. Coral over there. You're turning the... Sometimes these corals have other small animals that are living right on them. All right, let's get a good zoom on this one. All right. I'm going to try zooming here. Whoops. Sorry, a little fast there. That was me. Sorry.
In 1 pixel. That's 8 down there. If you just close it, just close the left. I'm going to try and come around to the left. I'm a little unstable over there.
Okay.
I've been watching.
Is that okay? Yep. All right, we can move on. It's just an RNA. There's 12 and now 13.
What did they say?
This is just
What's the genus of that fish? Or it's called a rat tail. Yeah. I always call those fish rat tails. Sometimes they prefer other names, but
also called a grenadier.
grenadier, yeah. There's another coral over there. I'll take that and zoom on it. Are you okay there?
Oh, that's.
I'm moving your camera. Okay. How do I know when it's looking straight ahead? Or how do you know when you
Well, I gotta
adjust
look.
that?
So that's the thing. I gotta.
You gotta find something on the basket.
Yeah, I gotta align it.
Gotcha. Okay.
And that's why it kind of freaks me out. It's like whenever I'm flying and it turns like I thought the
sorry.
ROV was turning. No, it's okay. I just let me know if you can.
Yeah, okay. And I can line it up for you again too if I use it. Now that I just use the basket.
Yeah, that's what I'm gonna do.
All right. Okay, moving up the hill slowly. We're 1,860 meters. A few fish down there. Eel-like fish. Gotten onto a sedimented section here. Anything you see interesting?
No, so far so good. But I really thought that at the beginning there was really like a lot of life.
Yeah, yeah.
Because there had been something at some point.
Mm-hmm. And still is apparently. I mean,
Yeah.
the corals and the sponges seem to be happy here.
Yeah, there
I
was a
mean,
crab
filter
and
feeders.
shrimp,
Mhm.
so there's the-
Yeah, quite a diversity there
Yeah.
that we saw.
well, compared to that, for instance.
Right, yeah.
Yeah. Here, if you dig into the sediment, you might find things.
Yeah, that's like,
Worms. I don't know.
microfauna, right?
Yeah, yeah. Is that a... I don't know what that is floating there. Oh, that's a fish. Until they moved, I couldn't tell. Are these little shells on the bottom, or... What have we got here?
It doesn't look like shells.
No, looks like little stain spots.
Where'd all the rocks go?
Good question. Yeah, they're buried, or yeah. I wonder what happened to the what was the source of that lava flow. I don't think we saw it.
So if you think about the crust on it that was manganese oxide, that goes really, really slowly, like
It's
just
old.
a few millimeters per like 100,000 years or a billion years. So those are really old.
Yeah. Yeah, pretty much all the rocks we've seen are really old. How old that is, I don't know, but millions, yeah, millions of years old. This is not the neo-volcanic zone where you usually find very, fresh lavas coming out where the mid-ocean ridge spreading is taking place. This is away from the spreading axis. Several million years worth away in terms of spreading rate. the spreading rate on this part of the mid-ocean ridge, most of the mid-Atlantic ridge, is slow. 2 to 2.5 centimeters per year. A human way of saying, describing that rate is about the rate that your fingernails grow. Although 2.5 might even be slower than that.
Wow.
There's another one just up from that. Whenever we go by an animal on the seafloor, the biology group in the back, 2 or 3 people, talk about it and try to decide if it's something that they haven't seen before and needs more investigation, or if it's just sort of normal and common.
So I think we kind of come up to this nice big flat area right here.
Oh, there's like a
I think you can see a little bit better where we are on this map. So we kind of came up this steeper slope, and now we're on this flatter. So we did right here. So we're at 18.
so the sheet flow was on the steep portion.
This is the part of the flows that are exposed to this issue closer to these patterns.
Right.
So all of that is still exposed to the first flow.
Yeah.
He's going to jack this thing and talking about the contours on that so that we can see the contours across the
Okay. You talking about putting contours on the map, on the ROV map now? Okay.
Gives them a geology.
Mm-hmm. So let's see, we arrived here just over 24 hours ago, right?
Yep.
Yeah.
That's it. Multi-beam mapping, then Toyo, then IUV, and now ROV.
Yeah. So you sort of, time on a research cruise gets a little strange, but we came to this site, just arrived a little over 24 hours ago. When we got here, we immediately did a ship-based multi-beam sonar map. The mapping system on this ship is really excellent. It's new. It's like one of the top of the line multi-beam sonar system. It's called the EM124. It has a narrow beam to give you better resolution, and when we looked at the map that we got from our three multi-beam lines that we did yesterday evening, it was a big improvement over the previous version, which was using a good sonar system, but this one really works well, and lets us see features on the seafloor that you couldn't see with the previous map. Then when you go to the AUV map, that's like a whole different level of, instead of 25 meter resolution, you're getting 1 meter resolution, and it's capable of doing even more than that, just put it closer to the seafloor. We're mapping with the AUVs at 50 meters above the bottom, covering as much territory as we can, and we're trying to map as much of this massif as we can. We're mostly mapping with the AUV to the east of here, closer to the spreading axis. This is sort of the backside or the older side of the mountain, and that we're coming up here. Currently, just seeing a lot of sediments covering everything up. We're looking forward to getting up to the top and seeing what kind of volcanic features are up there. Possibly any signs of alteration.
Don't you dare pull my arms,
And since you're up here, when we finish this one, I'll take another water sample since we've gone up.
Yeah, okay. Like a piston or a drive? You did bag 17?
I did a bag because I trust the bags
Yeah.
more.
They work better. That's
Yeah.
fine. Yeah, you can do another one.
They're easier to handle as well.
True. Yeah, I agree.
This detailed data, it looks like.
There goes a shrimp. Like the way they swim, kind of like they're galloping along. Few stray rocks stuck into the sediment here. Quite a few fish, some anemones, a little sea star down there. I think, no, maybe not. Okay, so that one finished. 0055. Depth at the end was 1820.
Go ahead, Rich.
Ben, you guys happy with the shooting speed, or do you want me to?
No, it's just not much detail.
It's good, Jason.
Much past that. We try.
See, I'm glad I stopped with samples up there.
Yeah, we've got a sediment section here.
That looks coherent again on another sample.
Okay,
We should be coming up to a steeper section right away.
all right.
Just wait a second, I got locked out for some reason.
Got locked out of logging.
Yeah.
We're getting closer to a steeper section.
I guess better close that one.
Okay. Yeah.
And
Again.
actually, I need to move over and hit close on 17.
Oh no, are you free for 1 second to grab me a pen?
All right. And just so nobody tries it, I'll hit close on 16 as well. So we could try a piston. We're not really going to have any indication whether it's working or not because we can't see the flow. We don't have an outflow.
At least
Rich, you just updated your position.
we would get
We'll test it.
Yeah.
We'll either get a
Yeah.
sample or yes. So okay, we'll just go with piston 1 then.
piston one then. Okay. Let's just tell me when you start.
I'm just moving it now.
What's the volume you're gonna put? 700?
Yeah, we can try for 700.
Isn't
We
it
don't
what
need
you
that
put-
much, but we do.
No, but wasn't it what you put in that last time? Or it didn't work?
For the piston, yeah.
Yeah.
Yeah. Okay, both of your RNA samples are done. So. Yep. These DNA samples can be used for a number of things. The way we collect them is just pull water through a filter, and it filters out everything above 0.22 microns in size. And there's a preservative that gets added to it passively. It's a reservoir that's connected to the filter, and it adds this preservative called RNA later, which preserves DNA and RNA to allow you to do analysis of either of those. And you can look at microbes suspended in the water, plankton. Do you get virus information off of those filters as well? The 3000.
Viruses that are infecting their hosts, we can
Okay.
detect, but viruses go through the filters,
Yeah.
unfortunately.
So there's a lot of work on viruses going on board the ship. That's sort of one of the main research interests for the microbiology team right now. Viruses are too small to be filtered out by this kind of filter, but they have infected some of the microbes that are being filtered out, and so you get some information about what viruses are infecting the microbes.
So I started typing.
Yeah, good. Yeah.
Seawater. Just
And while we're headed up the slope, we're going to take another water sample.
Position update.
We have different types of samplers that we're using. All right, we ready?
Yep.
All right. Starting that one. There are 16 water samplers connected. Actually more than that, but there's 16 regular water samples connected to this sampler. 8 of them are pistons, and 8 of them are plastic bags that they're put inside of a cylinder, and we pump the water out of the cylinder, and it pulls the sample water into the bag. They're pretty easy to deal with. As a sample, you just pull the bag out, close the valve, keeps the air out, keeps the sample in. You can analyze dissolved gases and most dissolved components in those type of samples. Then we have some pistons, which are designed more for really high temperature water and water that has high gas content. For the bags, if you put they can handle hot water because it gets cooled off quickly, but if you put a sample with high gas in there, it decompresses and expands when it comes up and just breaks the bag. So the pistons can hold the pressure in. They've got pressure relief valves on them that we can set up to 100 psi. You can lose a small amount of gas from that sample, but most of it gets kept in. Some areas of white staining in the sediment.
Sonar.
Just fast the camera.
Yeah, let's try it. Can we have a quick look at that camera that does the flow meter?
Okay.
Alright, that's turning, and I don't know if we can see any shimmering water coming out of the one next to it. Maybe. No, can't see it from that angle. Okay, that's probably good if you need to turn it off. Okay, lots of sediment. Not particularly fun to look at.
We're getting out of the gentle stuff soon.
Is this exciting or what? Yeah.
It makes the other stuff even more exciting.
Yeah. Wandering through the desert at the moment.
It's only when you've done such exploration that you really fully appreciate when you find this exciting site. I've been through 10 dives like this.
Yeah,
I
yeah.
need something.
Even
Yeah.
a lot of girls at some point are like, oh, hello.
This is part of exploring. You have to go and actually look and see what's there, and it takes time.
Yeah.
Okay, there's something. Well, there's lots of interesting ripples in the sediment and variation in color. What is that? Just a few
Every little
rock.
rock that animals find to live on.
Yeah, animals do not, well, sessile or animals that live in one place do not like to live on sediments because this sediment shifts around and they need a place to anchor.
She called them brittle scarps. Here we go. That's a rock. It's one.
Okay. Oh, one.
What the?
Oh, two, oh, four. And our depth was 17.
It's like indurated talus.
1780. 70.
Sample 5?
Sample 5.
Sample 6 now, right?
Yeah.
I have it.
No, if it's on this, like, it's didn't take all my information. I just took it if it's simple. I don't know why. Sometimes it works and sometimes not. So I guess this is
Do you want to just leave the?
can I go back?
It was 0037 to 055.
Oh, okay. So I'm sorry. It's just like this.
Oh.
It's I don't get it. Sometimes it just doesn't take all the information. It's
Okay.
I'll just put it.
Okay. Yeah. 0037 to 55.
So 37.
That was the RNA filter. Is
Yeah.
that the one you want? Okay.
So 55, huh?
Yeah.
Yeah, I remember that. I don't know. So there is a
If
main
it's
tree.
possible, could we get a really close one of those?
There is a main tree. You see that? And there's something I'm putting it.
Like the ones up here?
Yeah, that's fine.
Can I do it?
I think I want to see one that duration and fixing up the.
Okay.
I'm zooming.
But I don't get it. It's just like I was entering everything, submitting,
Oh.
and
Sure.
it
Thank
just
Whoops. Sorry. Okay.
Thank you.
Is that close enough for you? You want to go way in?
If you could get way in there, that'd be
Okay.
perfect.
All right. Get the texture there.
Yeah, that's great. That's perfect. Thank you so much.
We good?
Yep.
All right. Grab and go video. I guess we'll see if we're. Is that straight? Wait a minute.
So the other way you can do it is find like a feature like that rock. Is that
Yeah.
identifiable? And try and make, because this one's always centered.
Okay.
You can make that one try to be similar.
I'm not sure which rock that is. Oh, there it is. Okay.
It's a little to the left.
Okay.
So you're pretty close. I think
Yeah.
you're good.
Okay.
Okay. Photo with the talus.
we have some Niskin samples on, and do we need to stop to take those, or if we're doing this? Is it, I mean, I'm fine if we do.
You're good. If you could find that one.
Okay.
Okay.
And, if you could follow my arm, basically, but we need to check the camera
Mm-hmm.
while
We don't have to do it right now. I'm just wondering if it's something we have to stop for, or if we can do it on the fly.
Yeah, you can find this one.
This kind of thing. Yeah. Okay.
Some of these blocks of talus look like they're kind of cemented together. We saw a block earlier that kind of looked like that. Ooh,
Am I
something.
fine to go back over there, or do you want me to do another sample?
Yeah.
Can I go back there, or do you want to do
Yeah,
another one?
I think we're probably done for now. Yeah.
I'll be just.
Okay.
This stack of
That
rocks.
doesn't look super comfortable.
No.
Thank you.
It doesn't look in situ, but it also doesn't look like it's just.
Yeah.
I think it's talus, and it accumulates in one place, and then it like gets cemented together, and then that block of talus rolls down the hill.
You want to poke it?
No.
No.
You can poke it, but I'm looking on the right, and I'm starting to wonder if we're gonna start to see some in situ stuff. I think it's like cemented talus. Yeah. But I think we're getting pretty close to something. Like underneath some of this stuff.
There's a one. I don't know.
It's getting like coarser.
What do you suppose cement is?
Probably just the heat from the lava flow itself is enough to cause enough silicon gel and stuff together. You start to see this kind of topography, this like undulating topography.
Mm-hmm.
Oh, we're getting flat again.
Well, we have to go up about another 700 meters in depth to get to the top of this.
So unless we, like, we're not at the bottom of a super steep cliff right here, so I think a lot of that talus is like pretty close to being in situ. Just like at the bottom, like it's like
Position update.
this, and I think a lot of that talus is I mean, it could also be just brecciation that's on the volcanic flows itself. Rather than just talus. Let's see if we see some more.
Yeah. And any shelf edge that it might have come off of seems to be buried.
Dave, is your post going?
Yes.
Will you just let me know when you turn it off?
Okay.
Okay, cool.
I might. I don't really need to turn it off. I can leave it on. But if you're going to take a sample, you can always check. Have you run a couple?
It was doing some like weird stuff where it was like reading super high when it should before it hitting the injection. So I
Yeah.
had to push it out a bunch. I started injection so that it actually added some water to it, but it wasn't like for reading, so I had to like retry that. So I have it twice.
Okay.
So yeah, I'm doing one, but no. Not really getting it.
We should log when you're doing a reading. Is that going in C-Log?
Yeah, she's doing
Okay,
it.
you're doing that yourself. Okay, cool.
See, I'm outsourcing a lot of the. I'm outsourcing a lot of the logging. Making people do it themselves.
People should log their own samples if they can. Gets all the right information.
Mine is with everything.
Yeah. There's another fish swimming by. Eel-like fish. Jeff has just come in with a map on a USB stick that we're gonna install in the computer and put on our screen. We've seen the map, so we know what we're going over. It's nice to have it on the nav screen in front of you. So if you've been listening for any while, you probably already know this is the first scientific voyage for the new Falkor 2, Schmidt Ocean Institute's research vessel. And we have a group of scientists out here from NOAA, Pacific Marine Environmental Lab, University of Washington, Oregon State, Woods Hole, University of Toronto, University of Victoria, Monterey Bay Aquarium Research Institute, and French scientists from Ifremer. And we have the main mission out here is to try to find hydrothermal venting from serpentinization on oceanic core complexes. They are not an easy thing to find. There are not very many examples of them around the world. The first one that was seen is called Lost City. It's not far from here. It's on the Mid-Atlantic Ridge at 30 degrees north on the Atlantis Fracture Zone. And it has spectacular 50 to 60 meter tall carbonate chimneys that formed over tens of thousands of years and are still growing today. Amazing sights to see. If we found another thing like that, that would be really exciting, and I'm sure we would have a lot of other researchers who would want to follow up and do more work on that, because we just don't have many examples of that type of venting. But there could be other forms of that type of venting, lower temperature, different morphologies. Overall, we're just trying to find out how common is that type of venting on these oceanic core complexes. We don't really expect to see it on the dive that we're on today. It would be very sheer luck if we happen to come across something on this dive. We're going about it though in a systematic way using the AUVs to get really high resolution, meter scale mapping of the seafloor, where you can see structures that you can't see with ship-based sonar mapping systems. An example that we just went through working at another site south of here, it's a volcanic site on the Mid-Atlantic Ridge, right on the axis. It's a seamount or volcano called Puy de Fol at about 20.5 degrees north, and it's much smaller than what we're trying to map here. It's about 7 by 4 kilometers in dimension, sticks up about 100 meters above the background ridge, and and is shaped an oval with a flat top, and it's being tectonized and broken up with lots of fissures through it. There is one area of it where there's some relatively fresh lava flows, but then there's a lot of old lava in it as well, and it's got many areas of extinct hydrothermal metal sulfide mineral deposits, chimneys, or what used to be chimneys and have now fallen down into mounds of sulfide debris on the seafloor.
Would you be looking to zoom on that right yellow thing?
Where's
At the bottom of the screen? Yeah, it was a little bit more down.
if we can,
Yeah, right
yep, sorry, there we go. That thing on the right,
Yeah,
or
okay.
okay,
That was a crime, but it's not. You can continue. Thank you.
all right. Let me get that again. Oops, too far. All right.
Yes.
Anyway, to continue the Puy de Fol story, we're doing the same strategy here as we did there. It's just on a much bigger scale here at the eastern Mid-Atlantic Ridge Kane Fracture Zone intersection. So at Puy de Fol, we were able to map most of the surface of that flat top volcano with two individual AUV dives that were done simultaneously, launching both the AUVs one right after the other. When we got the data back from those two dives, we quickly downloaded it. The AUV team did, along with Jeff Beeson. The AUV team is from Monterey Bay Aquarium Research Institute. Dave Caress, Jenny Paduan, Randy Prickett, Jordan Caress, and Eric Trauschke. Eric is the operations leader for the AUV, and Dave's sort of the scientific leader for them. They have amazing capabilities with those two AUVs. They produce just incredibly detailed maps, and you could put sensors on them as they're flying their mapping patterns 50 meters above the seafloor. They've got sensors that we've added for what's called oxygen redox potential, or ORP. Detects changes in how it's not just about oxygen, but it's about if you put reduced compounds like hydrogen sulfide or dissolved iron or hydrogen into the water, this simple sensor detects those and gives you a deflection in its signal, which is just a voltage output. It doesn't tell you.
The coral pass.
Yes,
Stringy one, that big long stringy. Yeah.
you need a zoom.
If that's possible, that'd be awesome.
What is it?
So it looks like a black coral.
Black coral.
I'm not sure if it's species, but so we got one more photo and then we're good.
Okay.
Awesome, thank you.
Thank you. Okay, that's pretty good. See various things in the basket there. The intake nozzle of the fluid sampler and a scoop bag and our rock boxes that we keep the rock samples in. You want to zoom that?
Yeah, I mean, it looks like a manganese crust. Well, a lot of the things that we thought might be. Yeah, that's a pretty good crust.
Sorry, a little fast there. The dark black coating on those rocks is probably a manganese crust that forms over thousands of years, hundreds of thousands. Oh, you can see a bunch of spicules there as well on the ground.
What are those dark spicules? Around?
Are you thinking rock sample? Or
It's been a while, so I
mm-hmm.
don't know, maybe we can grab a piece of that crust. I'm kind of curious about these manganese oxide crusts.
You want, well, that's a pretty big one right there, isn't it?
Not the rock, the,
Yeah.
do you see the, like, flat lying stuff at the bottom? It
Oh,
probably
that,
looks right
okay.
though.
Okay, sure. Alright, so I guess we're talking about breaking a piece of that flat line.
It probably won't survive. If it doesn't survive, then just a piece of the talus will be fine.
Okay. I was talking about the ORP sensor, which has to do with what's called redox chemistry or oxidation state. And
For sampling,
See this little like loose? Yeah.
and we're talking about how to sample this piece of what we think is a manganese crust. Those grow at a redox boundary where there's reduced conditions below, that is no oxygen or very low oxygen, and manganese can be dissolved in a reduced redox state, and then when it hits the interface with seawater, it gets oxidized, so it can be migrating up through sediments and get oxidized at the seafloor by the oxygenated water, and it forms slowly these manganese oxide crusts. If you put reduced manganese up into the water column, that also would produce a signal in the ORP sensor, as does hydrogen, hydrogen sulfide. Methane doesn't really give much of a signal because it's not reactive. It is reduced, but it's not reactive. But the ORP sensor is what we have on the AUVs and also on our CTD package, and there's one on the ROV as we're driving around too. Alright, we're trying to pick up this rock with the manipulator or break a piece off of it that we can take. The manipulator has those red rubber flexible but grippy fingers on it, which allows you to pick up really delicate things. This stuff just disintegrates into pieces if you push on it too hard. So a bare metal jaws.
Can we try a bigger piece? Yeah. Look at that. It's so... yeah, it looks like indurated sediment. It's not altered rock. It's like the sediment that's all cemented together with the structures on top. Yeah. I guess the question is how coherent it is.
So we're seeing a thin manganese crust on top of sort of hardened sediments.
So what we thought was altered rock in the first sample.
I'll figure out a way.
Gonna try to lift up a big piece. Oh, there's a nice big chunk.
Oh, we don't need that.
Now it's too big.
You scared him.
There's a fish.
Oh, sweetheart, he's like. Okay, I don't think that we'll be able to get something that survives, so just maybe that rock.
You don't want us to break that piece?
Oh, you can try, but I think it'll probably still crumble.
I mean, if he grabs right in the center, it should try to fracture.
Yeah.
Maybe that's
You- You want to
Yeah, give it a try.
drop that whole piece in the rock box?
I think that will break apart if you drop it in, but
Yeah.
let's see. Let's see what happens when he tries to pick it up.
You want her to break it?
Um,
Can I give that karate?
yeah,
chop?
in half would be okay. And then if you can get the half that has a little bit of rock in it.
This is karate
Okay.
before.
This is scientific poking with the rock.
Ben's gonna karate chop the rock. Okay, there we go.
Yeah,
Now we have various sizes to choose from.
something that will fit in the basket. The top piece is fine.
Top piece is good.
Yeah, if it survives.
All these tips.
Okay, we trimmed that rock to a slightly more manageable size. And well, it's a bit more stable than some of the other things we tried to sample at Puy de Fol.
Yeah. It just turned
Which
into
were just
dust as
powder.
soon as you touch it, yeah.
A little bit closer, I think.
After this, if we have a minute to grab one of the bigger chunks of rock that's sitting around, I would like to take a look at it.
Do you see
Yeah,
an example in this other camera?
Yeah,
With it?
like that one right there. Something a little bit bigger would be nice, but just.
There's a, if you look in this one, there's a whole bunch of
Yeah, I knew this.
choices. Okay.
Let's put it a little bit forward. Try and reach a little bit closer.
Would it be more easy to recover this if we put it in a bio box versus lock box, or do you think it doesn't matter?
I don't think it matters.
The bio team might not be happy with me putting that way.
I think it'll make it in the rock box. It's not
It
much bigger. It's not much bigger than the jaw, so.
Yeah, it seems like not
It looks
too
pretty big
weak.
for the
you know.
small
There you go,
division.
It's fairly.
If it's too big for the rock box, then one of the bio box halves maybe. Look at that. Oh.
It's got a bump on it there that's making that hard.
Take the big and move it. Get a little bit deeper.
Anyway, as I was saying, the manganese is dissolved in a reduced state in the sediments where it's anoxic, migrates up to the surface, and then gets oxidized and turned into manganese oxide solid.
Ooh,
Oh, that's a pretty good.
nice.
This piece is actually holding together pretty well.
John, you can look that up. Can you spin it around?
Manganese oxides have a lot of other trace metals in them. They are related to manganese nodules, which take a different shape but similar chemistry. manganese crusts and nodules, especially, are something that's great interest for deep sea mining. There are really large areas of manganese nodules in some parts of the ocean. And when I say big, I mean deep abyssal plains of the ocean larger than North America, where there's manganese nodules all over. And they sit on the top of the sediments. They take a very long time to form,
Any other parts of it?
many thousands of years.
I don't know if the rock box divisions are big enough, so maybe
It might be, you can probably get it at an angle, and they have a little bit of play.
it's going to be very delicate. So maybe in half like the bio box B.
See right here? See how he's vertical right there? But I could put it.
Bio box B is big enough, I think.
This is so cool. Thank you so much.
Alright,
It's
so
just
open valve
very
up.
delicate.
We've got a rock in, but you want the one on the far right. There's a rock in B.
There's a rock in it? Or-
Yeah, there's a rock in A and B in the first box.
So we go valve box? Or we go valve box or rock box?
Wait a
It's
minute.
fine because I
Sorry, I may have gotten mixed up. Are we doing BioBox then?
I think it's just safer to get it in the bio box instead of crushing it up.
Okay.
I'd like it to be as intact as possible by the time we get it up.
Oh.
I don't want other stuff landing on it or it crushing into the box.
There's bio on it. It contains a
There's,
virus.
yeah.
Drop it here.
Yeah.
Drop it. You're on the lid. You're on the lid.
So this is bio box. No, those are others.
Looks small with it. A little bit lower so we're not dropping it.
They don't even have bio box. And it did break the thank you then.
Nice.
And another chunk of rock if you have time.
Just a loose rock. Yeah, like any of these loose rocks that are all around.
Well, we just broke.
Fist size, or what do you want? Fist size, 2 fist size?
Yeah, like a big fist.
This one right
Yeah,
Can
I
you put
wanna,
the lasers are, what the lasers are on right there?
what about the one below that, the slightly bigger one?
This one or,
That
Yes.
one.
alright, target, right for the lasers
Well,
on. That one's in place. You can bring the portion, bring it up where it, over that. Over there. And then you can pull it over slow and bring it up.
Okay, working on picking up this rock.
I got a little bit more pressure there.
Yeah, it looks like it's got a manganese coating on it as well. Makes it hard to tell what the underlying rock is. That sediment is just full of those spicules, needle-like, biogenic silica material. Yeah, you can sort of see that rock is coated. Got a little hollow on the bottom side.
Which rock box do you want it in?
Might as well keep
2C.
going and see.
Alright, so the middle two right behind that.
can I take a couple pictures pictures first? Can you zoom in on the rock and then not too far? Just yeah, that's good. One second.
You want the lasers on it?
Nope. And can you rotate it 180 degrees?
Price is right.
That is good. Go ahead and drop it in.
There you go.
C is the one.
No, I'll show you. It's this one right here.
Okay.
All right.
Beautiful. Thank you.
Anything else y'all need from this area?
I'm good. Bio team, are you good?
Yeah.
Everyone's happy.
All right. On we go.
Mark that sample.
What sample number are we up to now?
8.
The next one will be 8, or is that
This one is
that's 8?
Okay.
Wrap it up.
1,630 meters.
Can I request a zoom
On that sponge?
on that yellow stalked coral we're currently going by?
Yep, I see it. Come on. Let's go over here.
Yeah, that's the one.
Wow. That's just. Is that it? It's very skinny.
That one, but there's another one a little bit lower, but I'm going to take a photo of this one first. Okay, that's good. Take a move down, just one rock. There should be a bigger one right there.
There it is.
So I'm thinking that here, writing down latitude location, it's not that helpful. The time is okay, description is okay. Location just has sometimes.
We expect that'll be a little bit better.
So, for the next one. You want to do some. Yeah, I'll suspect it probably is. Do you have the computer that you can look up? And so the other thing I'm doing is I every so often go to the on my analysis group, and I just on until I measure right at 3 and we're at 1630. So we're doing a straight line between those two dots. So like to just put somewhere in the middle. That's great. Thank you. That's way too
Hold
close.
on, let me get that focus
Right.
for you.
Try to find our location
You are
relative
crazy, he said that's crazy.
so that I can follow along easier. So that's been my process. The contour thing is great.
Well, what do you think about that one?
Oh, stunning, stunning. Thank you. What is it?
There's also something for like the right corner of it too.
There's an expert in the chat that says crinoid
I don't know about it.
or a black log. Black log.
You said to the right of this guy?
Yeah, if you just go up slightly and go to like the right corner of the screen.
Yeah, right, to the constellation
white guy.
of stuff.
I see a brittle star. Yeah. What else are you seeing?
Oh yeah, there's a little brittle star.
Look at how much biomaterial there is.
Yeah, it's been like
All
that.
of this sediment is
It's been all through here.
nothing looks pelagic.
Little productivity. No spring.
Good?
Yeah, that's great. Thank you.
Are we on the move again?
Continue up the hill.
Okay, continuing up the hill. I was talking about manganese crusts, manganese nodules, redox chemistry, manganese diffusing up through the sediments and getting oxidized at the seafloor surface and turning into manganese oxide and co-precipitating a bunch of other metals and minerals. And as many people are aware, there's a demand for rare minerals, rare metals, some of them found in manganese oxides. And so the giant fields of manganese nodules are a tempting target for deep sea mining. They just sit on top of the sediments and can be easily scooped up. But if you start picking up manganese nodules from areas the size of North America, it's likely to have some impact on the oceans. And we might want to find out what that impact is.
But with this much manganese oxide, it's hard to know. This is just a fast pass. This isn't like the final rock construction or anything. Sorry, what did you want? I believe that's another 5 coral.
It is.
Just kind of check box anything that applies. I realize that iron oxide and manganese oxides under like hydrothermal recipients because that's the way my brain
Do you just want little snap zooms on all these little corals and stuff that I see, or
I don't get to do every one,
like
but
this?
if there's like a significant one, like that.
Yeah.
I mean, yeah, that's great. That's the same species as before, so that's awesome.
Cool. I'll just keep those.
Yeah, so
We're at 1620 meters, up from 1990 at the start, headed towards the summit, which is nearly 1,000 meters depth. So we got a ways to go still.
Is that a Gorgia? Have you seen a Gorgia yet? Fracture Gorgia?
No, we haven't seen it.
Kind of looks like one from a distance.
Oh, nice.
Turn that light on. So there's a view.
Ooh, that's a nice one.
How long did those take to grow?
If you wanted it in focus, let me know, or
Yeah, focus on it more. If you could focus on it more, I don't know what that, I think that's an anemone on top of it.
we can just get this in ROV real quick. Right?
It does look like an anemone living in there.
And then if the lasers can be in the shot after the zoom
power on the camera by any chance?
Is it like a lower camp?
Or the Gorgia, yeah.
I was so close. I was kidding. I'm just zoomed in a mile away. That's probably why it's so hard. You got the laser that they, you want them over here though? Like up in it or down at the base?
Below it. Below it, yeah.
Right.
Don't want to ruin the show.
Lasers always burn the show.
That's good, that's great. Awesome, thank you.
What this look? Is the black lawn, is it spot, urchin spotters?
So that orange coral is coil. That's a black coral. So that's a black coral. So the skeleton that ties with the black.
We've got our pilot Jason Rodriguez now sitting next to us, or J-Rod, man of many talents. He's probably seen more seafloor than just about anybody in the room. Being an ROV pilot for how many years? You've been doing ROV piloting? 13 years of ROV piloting. He knows a lot of biology.
I only do geology in the car.
Oh, props.
As soon as I go faster, I get
Passenger side. Or the driver's
in your
side.
arm or something, I get really close to dragging over.
This is looking a little bit.
So when you see something that looks anything different, like change in size or something, again, just throw in an observation.
Pitching.
When in doubt, just
Yeah.
make an observation.
Just build it whatever.
Thanks for saying that, J-Robert.
Hello. Now that's the, what, Falkor?
See, he knows all the names of these corals.
Yeah.
So would you give these crust on
So metallic gorgia has a specific species.
manganese oxide?
I realize now that there's a Hasley associated piece that is that's not hydrothermal, that's not right. So that's what I'm saying. Yeah.
You want me to get a good image of it, like a good in focus?
Yes, please. So, fun fact, this brittle star is always seen with this species of coral as the associate wrapped around its branches like this. It's really, really cool.
If you couldn't hear the biologist in the background, there's a brittle star that you can see that's wrapped up. It's got its tentacles wrapped around the coral and lives together with it. And according to them, this species, these two species are almost always found together.
The number of injections matters. Beautiful, Jared. I think you're so pretty. Wow, that is sharp. Sorry, all ROV team, not just J-Rod.
Yeah.
No figures.
Is that good?
Yeah, gorgeous.
great.
Thank you.
All in the room?
The brittle star on the metallic gorgia is Ophiocreus edifice.
Ophio what? Ophiocrius.
Ophio, yeah.
Okay.
Oedipus.
Oedipus.
You have a coral expert in the chat.
Is that Jeremy?
Steve Ostrich, it's another insect species.
Okay. Yeah, that's a cool aspect of this live streaming is that there are a bunch of great experts out there in the audience who can follow along and tell us what we're seeing, because some of us out here, when it comes to biology, especially me, don't know these species at all. I'm used to looking at hydrothermal vents, so I can tell you something about what lives there, but I can't recognize one coral from another.
You better be. Okay, this looks like we're getting in situ some pillows.
Yeah.
Can we just slow?
There's a pillow basalt, an old one, cracked up.
There's another coral on the right.
Just another coral on the right.
Curious, but David,
We-
if you have time for filming.
Well, we're making fairly slow progress. Oh, there's one there. Okay. That one also has a brittle star living on it.
Yeah.
It's all good. Thank you. See, ideally, we could have
Another laptop.
That's great, thanks.
You're okay.
There you can clearly see those two species living together.
Am I going back to the pillow lava? Am I moving up?
Moving up, but we're looking for something that looks like it formed right there in place,
Maybe
like a lava flow that we
Point
can
towards
sample
the
at some point.
pillows. Well, I don't know how wide that is. That looks like a pretty big flow section we're looking at there.
They seem to like that.
If there's any
Quite
of those
steep over
pieces
there.
of rock right in front of us that you can grab, then I'd like a sample.
Say that one more time.
If you pan back to the right there.
Yeah, I was just giving this to you
Oh,
first.
I want
Yeah,
that.
yeah. You want what do you want?
Just hold on 1 second.
You change your mind?
No.
She wants a piece of one of these pillow basalts.
Yeah, so there's some of the small rocks right in and around it. Yeah, kind of same-ish size as last time. This is the first like convincing in situ stuff.
Mm-hmm. It's a really steep edge of a pillow mound.
Can you speed up port 1008? So slow. So slow. Alright, so she wanted to use
Okay, looking at these old pillow basalts covered with biota.
Looks like you're doing all right here.
Oh, yeah.
I feel with the conference with any one of you guys.
I'd be happy for you to sit down here. Huh? No? Okay. When you're ready. We're taking a few water samples as we go along too. But we don't have to stop, we'll just do
So
it
you
as we
just
go.
take them,
That piece right to the left might also be easy to grab.
Yeah.
so you're just taking the wand in the holster?
It's just in a
or-
holster, yeah.
Yeah, okay, and then you just use the pump.
Pump's
then
running.
you- yeah.
Pump's running all the time. Oops, that's too late. I probably should
Oh, you
turn
wanted
this
the
off.
bio? Do you want it in a bio box? Can we put that in a bio box and try to collect that bio too?
Sponge.
Alright, quartz is coming out.
It's a geo sample. That's pretty popular.
Alright. Bio box is coming open. Into a zoom. Alright, and let's go with the other one, or we just go separate, right?
Separate, please.
Roger that, so you gotta go.
Rock, you can see the outer crust layer on that, bunch of oxidized material in the underlying part that solidified more slowly. The pillow basalts form a crust rapidly as they're extruded like toothpaste out of a tube, and the outer parts freeze while the inside remains molten, slowly cools. Lava keeps pushing from the inside and breaking through opening cracks and pouring out as another pillow, one after the other. Sometimes they form very large mounds. This one seems to have erupted on a steep slope, and one pillow follows another. This is something we've seen happen in real life at West Mata volcano in the Pacific. Yeah. In 2009, we were there when it was erupting and saw pillow lavas forming at, I think it was 900 meters deep.
Now that we have this formation, he would like to try DNA and see if he can detect them.
Oh, yeah, can we take a Niskin here?
One of the big.
Sure.
Like, right in this exact location?
Yeah, right over all these sponges.
Roger that. So, Adam, that definitely worked.
Is it a little too fast?
No, that's perfect.
Just note, four doesn't have this extra flag on here. So with the flag four, we can see
Alright,
the-
which one are we going for right now? What comes up? Those sheets.
Oh,
Anybody got a sheet?
sorry.
Sorry. Really?
Which Niskin?
Yeah, which list comes up?
They're all open. None
They're
of
all
them have
open?
been used. None of them have been used. You want one?
Yeah, that's right.
Sure.
Thanks, sir.
So should we do Niskin 1?
Yeah, Niskin 1. Why not? Start with 1.
I can log it? Yeah.
Is this for Sean for eDNA?
I'm logging it for you.
Is Sean in here? No. But he wanted one for eDNA.
Yeah, I went and talked to
Do
him.
you just
okay.
want 5 liters? Do you just want a 5 liter Niskin, or do you also want a 2.5 liter?
No, only one.
Are you gonna sample no.
I'll wait till they're done with the other samples.
Okay.
But yeah, I'll probably take another one kind of soon.
I'll just squeeze in.
Okay. So the camera right now shows the arm pushing on a trigger lever to fire a Niskin bottle. You can see up in the upper part of the camera there, it's got closures on both ends. They have a spring that pulls them together, and when we push this, it's supposed to release what's holding them, and they slam shut, trapping the water inside. And as we look up, yes, there it is closed, the one on the ends. That right at the top of the screen, that's the bottom of the Niskin bottle. So that traps 5 liters of water, and Sean McAllister is going to filter the particles out of that, then look for DNA to match to what's living on the seafloor. So this is what's called environmental DNA sample, looking at DNA that's just floating around in the water. Interested in using it as a quick tool to tell you what's living on the seafloor by sampling the water above it. So right here we can see corals and sponges and brittle stars and probably some other things that I'm not noticing, and their DNA would be in the water, and we take that and do the sequencing and say what's there. Simple concept, but devil's in the details. What sample number are we on now?
We just did 10 with this.
That was 10.
Next one will be 11.
Alright. Okay, I guess it's time for another water sample. So, let's see.
This is just HFS, not
This will be HFS piston 2. Filtered. Okay. I might just take a bag too.
Okay, just let me know.
Okay, there's piston 2.
Is this number of sponges normal?
700. That's a lot of sponges.
Yeah.
Super dense.
Oh, you see a tiny little ropey part of the pallets on top there. Very top of the screen.
Okay.
Got a really big possible metallic orgy coming up. You might want to see this guy. This guy's gonna be fun.
Is this like brittle starfish?
Looks like a dead bamboo.
Oh,
Yeah.
1590 meters.
Dead bamboo.
yeah, it is a bamboo coral.
Looks like there's
Okay,
some polyps
I guess we'll
down
start
below
that.
and scattered out.
Yeah, there's
Ready?
polyps
Yeah.
all over this.
There was a sea panda down here.
What's the red thing on the left?
We're at 11, right?
And a crinoid.
Yes, 11.
Who's
We're
the crinoid, the big fallen? No, the crinoid is the one
nobody
that
else cares. You're logging in.
Wow.
We don't have to tell them.
Oh.
All right, we got 209.
Feather stop.
That was 1590.
Okay. So the biologists are having trouble keeping up with this. talking, so I can go a little bit slow for this part. Can you start talking?
Yeah, I can definitely do that. I got something to go faster.
Yeah, we just go slowly and just keep moving. It's okay. You guys can also like just do a bunch of photos and then log it afterwards.
Yeah, that's what I think we're gonna do.
Just be like start
There's too
a
much.
yeah. So just take a lot of photos with nothing else. And just put them to be. I mean, we're taking photos every time.
Oh, there's
Wow.
a shrimp.
Yeah, that's a nice one. It's got a brittle star on it.
Do some scripts up in there.
That's a nice view.
Language.
Wow, it's so cool.
Look at that. That's a nice brittle star up there. That's the star.
They like pillows too.
Kind of cool. I might have to just take a picture of that with my phone.
There.
But once they start asking for stuff and you know what it is, it makes it easier.
Oh, that's nice.
Brittle star wrapped around the coral there.
Trying to catch as much as it can.
Trying to catch his what?
As much as it can.
Yeah, gets suspended up above all the other things.
Yeah.
Do you want a sample of one of these things? Do you want to try to get another rock with some sponges on it, or like that rock right there with that two level? You could probably grab it.
I think there's a lot more coming up that y'all might want to see. Yes.
I think you should trust J-Rods.
There's a lot of dead stuff coming up here, which normally means that there's a lot of live stuff coming up above it. That's up to y'all. Y'all tell me. So,
Oh, it's now.
yes, starting to get a lot of this dead stuff.
Do you want to monitor that? We already have a rock. Oh,
There's the one I'm looking for.
There's a big one.
yeah. Another rock. Yeah.
Dun-dun-dun.
So I was probably gonna filter.
Big coral coming up.
Can we grab a rock sample with a bunch of biology on it? Whatever we can.
That sounds very easy. It's all over the place.
Yeah.
Wow.
Like that. I guess we don't want to pick up the corals, right? We wouldn't want that.
There's this one.
I think we would want, but we
Too
won't.
big to fit in the river. No, but we can put it in the bio box. The other one is on the sponge too much. Oh, look at that.
There's an urchin, yeah.
How deep are we?
Sponges.
1570. Oh, that's a nice one.
How are we doing? We're still going.
Yep.
Okay.
Look at this. Look to the right. Look at how vertical that is on the right.
Yeah, it's really steep here.
You want a beauty shot of that?
Yeah, that's a one.
Yeah, that's a nice one.
Yeah.
It's moving.
Mm-hmm.
Beauty shot of that.
Thank you.
No, it's for Monica. I still want a beauty video.
What is this crusty stuff on the rock there? Is that all biological?
Encrusting sponge.
Is that all just dead sponge? Dead sponge. It should also be different species. That's another crusty sponge. Because it doesn't look like if you look at the ones with the baby crab. Baby crab on the rock? Up higher? Oh, it's moving.
Starting to
Let's keep looking for a bio rock.
Bio rock.
Just opportunistic if we see something.
Something cool.
Oh, there's also, oh, what is this? What is that?
Anybody want to see
Sea-pan.
What's that? That looks maybe like a, what is it?
It looks like a sea pan.
Yeah, or like a rock pen is like what they would call it.
Somebody in the audience will probably tell us. Are they?
We can keep going up and
Roger, going up, looking for a bio rock.
what is that white thing there? That looks like a mushroom.
Sponge.
More sponge, different kind of sponge.
Got another dead bamboo coming in.
Okay.
Sea stars eat. I'm not sure how they all like that. That's why I call them sea babies.
How you doing?
Good. You?
Me? Tomorrow, you?
You want to sit here? Yeah. I need a break.
Oh, we're done.
Well, wait a minute. Okay, just record this sample and then I'm going to get out of this chair.
When did they end?
209 to 215.
Okay, and
700 mils. Huh?
three.
It's subvertical. We don't have like a number or anything, right? Like 80 degrees.
Please tell me that it worked.
Monica, if there's something you want to get, just you see a rock with all the biology that you think is about the same size as the last one. Well, back down below.
Yeah. Look at these feet forward. That'll give you an idea of how
So it's about so if you see one that you're like,
I guess that's probably enough for the moment. I mean,
Okay.
I feel like it'd be a little more certain to take a bag.
Look that plan, so it didn't record what I did, which starts to get annoying.
Oh, sea lug's not.
Well, I don't know what with my computer. It's the second time when I feed everything and then just.
so you want a rock with sponges on it?
Yeah, that like one that you
Yeah.
one of those would be perfect.
Okay. So we're looking for sponges on a rock, J-Rod.
That's pretty easy. There's sponges on almost every rock.
As big as you can get it.
Challenge.
Or a rock with like multiple.
Challenge accepted.
Multiple
and
sponges on a
acquired.
rock. Are the lasers still on? Yes.
How about that?
Okay, well,
You want to
maybe I should take a bag too, just in case
Yeah.
that didn't work.
Could you just
Another
give me
Gordia.
back the time?
How about
because
this
it's
one right
already-
here?
209 to 215.
Okay.
Yeah.
Yeah, that would
2:15.
Yeah.
Yeah. And time.
And we can put it in bio box 1A with the other one that has the
Oh,
sponge.
it's written there.
Yeah, it's still
We're just
there.
gonna have to make sure we have some.
3 temperature.
Identify our rocks.
Max temperature.
That'll be good for
There's all these
Now, what love?
all this biogenic debris through here.
What do you got?
There's a tiny little squat lobster, pink one.
Okay, so they want to go for this one right here.
yeah.
Cool.
Okay. Let's hope this time it
Yeah, we'll
works.
make it pointing at.
All right, so I'm gonna go to a bag. I guess I'll. Let's see. Filtered
just-
all the way through 19 is filtered, right?
Yeah,
I'm just gonna
17
do-
is
it's gonna be
HTTP,
23.
so unfiltered bag.
Yeah.
Okay. Do we have something on 23? On 6?
16 is not no sample, but
You want the one with that
23.
big
Yeah,
sponge,
okay.
because there's a rock, small rock, it's attached to it.
Do you want the big sponge? No, the smaller sponges.
Roger that. Small
Spare
rock
it,
it is.
spare it.
Just tell me when you start.
Okay, I guess it's there.
Starting now.
Okay.
And we're at 1573. 73 meter.
We were 11, so that's 12 now, and
Bag 23. Started at 220.
Where do you want this to go?
Bio box 1A.
1A?
Yeah, with the other one.
It's gonna have a frame. Right, Box is open. Box is open. Do you want a picture of it?
Yes, please.
Sorry, that's kind of, I just thought I'd
Yes.
better finish one last fluid sample and then I'm gonna get out of here.
It's all you, man.
I'm enjoying the show.
Okay.
It's
take your
bio show.
photo.
Is bio show?
Okay.
Can you rotate at 180 degrees for one more photo?
Do it over the, come over here. Get over the bottom. And it's going into A, so the left one. So, yeah, arm down.
Wow,
Alright.
delicate stuff on there.
Yeah. Makes it to the surface.
Yeah, be difficult.
Yeah, right there's good. Do you want, or is this the,
One thing that sample can be good for that I can think of is that now Sean, who's working on the eDNA, has an actual sample
so you could do,
of the dominant type of sponge that's sitting down here. So he can compare that with his
I mean, I don't
water
know.
DNA.
No, because it's okay, yeah, I'll leave it. I'll leave it. You'll have to just come up
And there are other things on there as well.
I will leave that.
Okay, in the box. The bio boxes have sealing lids, keeps everything in, and it also keeps it colder as it comes up.
Moving on it?
Yes.
That one's almost done.
Okay.
222.
I hope it works this time. Yes, it worked.
I don't technically see this actually. It's too close. It's so cool.
Wow, so much stuff growing on all these pillow basalts on this steep slope.
Don't need me to go slower than that, or is that good pace?
We need to move. Got
Ooh, this is at what? Something 9, like I-9 or something like that? Here's the word next to that one. Adds a number. Looks like it's a Star Wars character.
yeah. This is a more biological dive than
We don't need
we
to
were
like,
expecting.
if you just describe the
All kinds of stuff growing here.
the point that it starts and what the point that it ends, and you can even like just revisit that part of the video.
I thought they were attached to it.
Do you want to talk about anything to the people out there? No,
I'd rather
not
not.
really.
Maybe when there's actual trees and stuff, I can say stuff.
Yeah. Okay.
But at the moment,
Okay.
I'd rather let the people that knows best.
We should have a biologist up here,
Yeah.
but they're too busy logging
Logging
the information
everything.
and taking pictures.
Like back
Which
off the
makes
wall so
sense.
they can't see it anymore. I'm just like.
can put Jeff in the chair, Jeff Beeson, our geologist from Oregon State. Or
I thought he was in Nebraska.
he's not from Nebraska. You're not from Nebraska, Jeff, are you? Okay, so I'm going to turn you over to Jeff, who will say something about the rocks that are underneath all this biology and what we're doing to understand the geological structure here. Okay, wait a minute, one more button.
No hurry.
There's Nebraska.
There's the what?
I said there's Nebraska.
Don't. I've never been in that state, but I'd like to go. But before then, let's get to the top of this hill.
No, let's take more samples before we get to the top.
Shoot.
Okay, no more samples.
No, hey, who put Jeff in charge?
I know.
That was a mistake.
Go, Jeff.
I like to get some meters under our belt.
Can we make sure the lasers are on, please?
Lasers coming back on.
Yeah, because we got
We're
a little less than halfway up this hill.
But
Yeah, but we're getting steeper.
distance-wise, we
Yeah.
have covered
We're covering quite a bit of.
that, so we only have like a bit where it's just gonna be like this.
Yeah, we got time. We're certainly gonna make it to the top.
So
I think we're gonna make it
in
to
100 meters or so, we can grab another rock.
Yeah, we can. Of course, it's fine. I was
Thanks,
joking.
thanks, boss.
Yeah. We get the samples that we need.
So it's looking more talusy now again here, like a little bit less coherent pillows, but we'll see if they come back into.
That's a technical term, J-Rod.
That's a fun word.
Yeah, talusy.
Oh, there's a pillow. We like pillows. So pillows and.
Probably before that.
We are looking for
We need to be
small
on deck.
of
By 6. Probably 5. On deck by 45. Just picking up the sides. Leave the bottom up. Which way over there? Should peek off to this side.
I guess we don't need much more than a hand. And which ones are the, like, what did you, which ones? So, like, not. Oh, wow.
This is quite the vertical cliff of sponges.
When you see those,
Some pillow lavas sticking out. Sponges, corals.
I'll give you a shot for outreach. Do you want to zoom in or?
Can we sample that?
What, the coral?
Yep. Into a quiver?
Just a piece of it?
Yeah, about the size of a hand,
Yeah. Oh, so enough that fits inside?
enough that fits into the, one of the quivers.
Let me,
So maybe just like a branch of it?
Let me get this for outreach real quick and then I will set up for that.
So that will
As long as Jeff doesn't yell at me.
492 bio 13.
I wouldn't yell, but I will speak sternly if needed.
Can we get the lasers on there, please?
Just give me a second. We're doing the outreach portion of it and then I will put them right back on the.
Doing the beauty shot.
That's an amazing channel to compare us.
Roger that. Lasers back on.
Incredible.
There's our lasers. Alright, I'm gonna get you set up to get a snippet of this.
Yes.
Can you introduce yourself and give us a little bit of info about your scientific interests?
Oh. Speed. Okay.
You're doing great.
Oh, thank you. So I'm Jeff Beeson, a researcher at NOAA's Pacific Marine Environmental Lab and an assistant professor at Oregon State University. And my interest is in geology and geomorphology, in particular at this area, the geologic evolution of oceanic core complexes and their interaction with hydrothermal venting. And this is a very exciting dive to get to the top of this massif that is a oceanic core complex in kind of in its infancy. I've never dove on one of these before, so it's really exciting to get to take the maps that we make with acoustics and get to see what it looks like in really small scale. Very impressed with the sampling dexterity of J-Rod and Sebastian. Oh, Joe's doing it. We got very experienced samplers going here. Are those lids attached? Yeah. Okay, good. We don't want those to drop. We're not going back to get them. Much of the work I've been doing on this cruise so far has been monitoring, collecting, processing, and doing some initial interpretations of our multi-beam bathymetry maps.
So the one closer to us or the one above it? That one I'm kind of...
The science team is trying to decide what to take here and put into the quiver. This is a delicate operation. Wow.
Way more, like if we got access, way more interested than I ever expected. Yeah, I expected just like some rocks and all this situ giant pillows.
Going in with surgical precision, operating a large robot 1,500 meters below the vessel in the middle of the Atlantic Ocean. What a world we live in.
Do you want to put that in a bio box? Yeah, put it in deep on top of the rest.
Nice and easy movements. They will grab on.
This is holding on.
He's holding it together.
Ooh.
No, don't drop it. Oh.
Just missed catching it with a basket, but we have some.
It's okay, they will. It's kind of sad that they.
Good thing about coral is they will catch on somewhere else and start throwing it.
It's just taking a little ride. He's relocating to lower down on the slope.
Let me bring that fortune. Get out of here.
Placing the sample into our quiver in S1. Oh, oh, grab it. Oh, I'm gonna slide. Just
There's a comment that the volume is low for the commentary. I got that from Susan.
that was a while ago.
That was a while ago. Oh yeah, that's it.
Come on.
You gotta talk about
Tell me why you're happy about that sample.
well. Yeah.
What's so exciting? We got Monica here. She was all smiles while that operation was occurring.
There's a scientist coming on the next cruise that is conducting a coral physiology study, and so we were able to collect the sample for them, and to also get my first look at a deep sea coral.
Wow,
in
that's
person.
awesome. Yeah, and look at that. We have our onshore scientists that are getting to see samples collected that will help them. We're doing amazing stuff out here. Thanks. That's really cool.
Are we good to keep going?
I believe we have been hanging out here too long, far too long. Let's get rolling, or floating.
We're happy. Y'all already put a lot of rocks on us. We're happy.
Yes.
There's no
Use
floating for Sebastian, right?
thruster us up.
Good job.
J-Rod, how do you know that we're not going to run anything into anything above us?
Because we got a whole bunch of cameras,
Yeah,
but
Are we looking straight up on a camera?
I have pretty close to straight up, so this one right here.
Oh yeah,
So
that's good.
yeah, I can get a good That's how I know what's coming up.
What's coming up? That's how. See, that's how he knows what's happening before we know what's happening. One step ahead. All right, we're continuing our ascent up this very steep wall. We're traversing 1,500 meters roughly from where we were at the base. We're at 1,540 meters, 37 meters water depth, and climbing.
Eating coral. So weird. Eating coral. Yeah, sea fish. Sea fish. Big old sponge.
You're saying it's gonna the really steep part. It's gonna be amazing. It's gonna be the overhangs. Yeah, that's what I was talking about. Worried there could be some overhangs up here, but we're doing it. We're looking for it. I know.
If it's a really bad one, we'll know because things are falling over.
If we bunker, then we know.
You can go ahead and get the bus over 139, maybe 45.
Yesterday, I was working on mine.
We fixed it.
What did you say, 100 what?
So we're 140 away. So just keep on. Yeah, that's perfect.
When you call them, they see it.
Yes, they normally do see it.
Only. I was just thinking that we have almost 500 more meters to climb to get to the summit.
What are you looking for?
Changes in lithology.
Something that looks different.
Different is good.
Well, there's some weird looking pine needles on the ground.
I've seen a lot of pine needles, although I've asked the biologist what it is, and they're unsure, although the sea urchin bits, maybe? Or suggest there's a lot of sea urchins above us, though. We haven't
Dead coral stalks, maybe?
possibly dead coral stalks.
We saw so much coral.
Urchins like to hang out like right around the edge of like bacterial mat areas, right?
There's little
That's what I've normally seen. Like
Okay.
I've seen like just like the mass quantities of
It-
the mountain sand.
It would be very exciting to get to the top of this and find lots of urchins around something like that. It's like doing geology on land. If you want to know what the mountains are made out of at the top, just go to the bottom and see what's fallen down. SuBastian is essentially free climbing a 1,000-meter mountain right now.
One of the scientists we had on here before described it as like going to Yosemite National Park for the first time to explore how beautiful it is, but you could only go at nighttime with a flashlight.
Right. Yes.
So the summit is opposed to 653 meters.
And we have to climb another 500.
So it's only
Oh, we made even a different kind of lava. What's this one coming up? Is that just a big pillow that's collapsed? Is this a...
That looks like a sheet.
Yeah, that's what I was saying. It's a different type of flow. Can we, do we want to pause?
Can we get a like chunk of the bottom of that?
Do you see this sheet flow? It's this unit here. It's not a pillow. It's more. Is there any blocks that are grabbable?
it looks like from the bottom. That's
Yeah, from the bottom.
the upper cut, yeah. Try to come way down so you can get up underneath it. Window. That's a window.
I mean, it looks like it's jointed, but I think we'll end up with a pretty big piece.
It's a lot bigger than I thought it was.
Yeah, it doesn't look like a nice little thin thing.
No, maybe we'll come up a little bit. And up a little.
So this is a new type of flow we've seen in this area. We've been mainly seeing pillow lavas. This is more looking than a sheet flow.
So if you can get that little corner piece, you're good with that?
Yep.
Yeah, give me a second, let me just land out. You got it, or you want to get it?
Get a big.
Oh, that's how it's working.
Oh,
Might be a better Brad, but
oh, gotta love the manipulator cam.
I can come around. I can come around a little bit if you want. Like about there. Yeah.
Stuck on there real good.
It's on there.
It's on there.
What you got? Try to look what you got. Nope. Yeah, I think we got that. So I mean, we still got a hold of it, but now it's not breaking. I'm trying.
Like I said, Sebastian is mountain climbing.
You didn't bring a rock saw on your ROV?
That
So if there's another piece that looks like it can break off, great. Otherwise, we can also just keep going. Like, what about, does it look like it's kind of coming up vertical here? Or is that just an optical illusion?
No, that's a lip.
It's a lip? Can we grab a piece of the lip?
It looks like it's.
Does it look like it's sticking up?
Oh, I see what you're saying. It does kind of, doesn't it?
Yeah.
Get my head a little bit. Get you a little bit more straight on that. So you just come down on top of it.
ROV great. Is that an update position?
Yeah, Jason, start making your way there, please.
Okay.
In the bottom right, like right below it. Oh, the picture to the left. Oh, well, I think so. I don't know. If we can probably put it in a quiver. Is it too big? How delicate is it? Yeah, come down. Do we have one already? Okay. Well, everything's possible. Okay, we'll see if we get a sample from the rock.
It's stuck on there too?
I can try to get you up a little bit.
The best rocks are always most difficult ones to sample.
It's on there. Well, I think we gave it a try here. The bio team has requested a sample of the that guy to the left.
Right here.
From the arm. Yeah, that guy. The whole thing, not the into a quiver, maybe. Or is it better? I don't know how big. I don't know what it's connected.
I mean, we might be able to tuck it in one.
What kind of sample box would be good for this?
You could put it in the
Bio box.
The other one, the bio box.
I want the whole thing. What's that? So you just want like right where the end of the basket star is?
I think it's gonna all pull off if you grab it. Probably all gonna.
The coral cutters will cut it.
Oh.
Yeah. So we can get
yeah, it's hard to get it like a sense of size.
It's not massive.
I assume you can't just grab a branch probably. It's not big enough for that. Can't really tell.
No.
You take the whole thing, I think.
I would think you have to go just to the right of the basket star.
Well, whatever you guys.
Give it a try.
I'm just going to end it real quick. So just get the red just past the.
Sean, are you logging this sample? No, this is sample 14.
Yep. 14. And then I know I just rotate the draw. Have to wait a little bit.
But you can. Yeah.
Rotate both directions. More than that. More than that. 90 degrees. And then the other way. Rotate 90 degrees. What you got? I'm good. Firebox?
Which one? Let's do Firebox 1.
A?
Yeah.
Roger
Top
that.
of the other stuff.
Go to A. So I'm using my plus right now. Then start rotating counterclockwise. Okay. Don't bring your arm towards us anymore. You actually go away. Bring your shoulder right. Shoulder right. Alright, y'all left. Y'all left. And shoulder right. You're all right. There you go. Now you can go down. Very good to release.
Absolutely beautiful.
We can continue our climb when you are ready. 500 meters to go to the top. Okay, coming up on some more massive pillows.
Is that a good seat?
Yeah.
Yeah, I'm just tracking the samples by number and the samples. Some more sheet. And a lot of fluids necessary as well.
That was a metallogorgia, right? Pretty sure it was metallogorgia.
Species? What that?
There's some more of that rock y'all were just wanting to get a sample of coming up.
Yeah.
Not
No,
that one.
keep going.
No, this one right here.
I'll just take a photo of it and we can keep going.
Okay, copy that. And there we go. Coming up.
Going up, going up. Continuing
Ooh,
our
that piece
climb.
right there, can you get that one right there?
Roger that.
That looks like it's doable. Jeff, are you okay with that?
Yeah, collect what we need. The top of the mountain isn't going anywhere.
Just right. Let me get back to where she went. So you see that piece right there with all the veins on it? Almost just lower center screen, almost middle center screen. Lasers are on it, right? So where the lasers are. She wants to stick on that.
And there's a shrimp walking around on it.
Alright, got a view on both cameras for you.
That was nice, like. I'm really happy to be doing such stuff. A little bit sad that it's mostly lack of things that we could have done the hill rather than section. Like it would be really cool to see some trees and it's like hikers and really just seeing that.
If you go in there, does it easily move? Let me know if you're about to start racing on it. So
It's very steep,
but it has to be the one with the ripple.
top of it. This is vertical.
The one with the ripples on it.
Or near vertical.
Give it a good push. Give it a good push. You can try it for a while.
I can take the if you want to sit there.
I can do the log. Or you do the log.
Tamara, do you want to sit here with the fluid stuff or here with the
That one's pretty stuck on there.
Really?
Yes.
Oh, how about just one nearby? Then?
One nearby. There's a little one sitting on top of it, but it doesn't look anything like it.
No, like just slightly bigger than one. Oh, the little one on top of it might be okay.
I mean, you want to just have a quick peek around?
No, please. It's your
Anything on here? There's nothing on the top of this one.
Go ahead and try, yeah.
You see what I'm talking about?
Some
I'll
bio.
let you.
Yeah, right.
We're gonna do the rotating carousel
Right there.
of scientists sitting at the chair. We have Tamara Baumberger. Gonna come on. I'm signing off for a few minutes. Continue up the hill.
Yeah,
Keep
that's my
going
job.
up.
Keep
That's
going
good.
up. Stop the bio people taking all the samples.
We did get a bunch of nice samples though.
You can't use mass as that rock had a lot of that's true.
We need to solve that.
So if you rotate clockwise a little bit. It just crumbled off.
Okay, just a piece from the seafloor around it that's a reasonable size.
Did that?
Something that you feel like you can grab, I'd be happy with.
Collecting rock samples here so that we can describe them once they're back on board. Also look at them under the microscope to get some idea about their formation history and also the composition. And once we are back home, they will also be run for their detailed geochemical composition.
Yeah, that one there looks good.
Trying to find the best candidate that we can actually pick up. It's not very easy to sample these guys, so it's hard to break them off. So we're trying a few options here.
That was pretty big. There's one right behind it that might be easier to grab. One of those guys, or even to the right, one of those ones. Or even further to the left, I don't know.
There's one up here.
Yeah, that would work too.
Look at these guys, right here in front of the fork on there. They're just nice humans.
Sure.
You know,
I'd like there to be some thickness to the rock if possible.
with a port arc? Absolutely. You got this stuff around here.
Gerard, we do need the sticky poker again. We do need the pokey stick again.
We tried to do this one, but it doesn't work.
Oh,
I mean,
oh yeah.
don't, like, if
Occasionally.
you're looking at the TV, it works. But
Yeah,
I
see.
can't tell, oh, it works on white.
It's very random. Yeah, if it works or
We gotta
not.
make
Yeah.
a new one.
Jara, what about, is that stuck in it or?
We will find out. The port arm, we can be a little bit more rough with, because it doesn't have those bells on it.
I'm trying to find the pieces of grab rocks, but I keep making decisions. It's like it should be.
Oh, no, the whole letters, because it has the whole letters.
No, we've had it plugged in years.
We're getting nowhere.
So we know that's
But we're not getting forward very much. We're going.
we've done like.
For this one, so you can't get it.
So we are now using the other manipulator arm. They are the same, but this one doesn't have the coral cutters on it. So you probably have seen before the other manipulator arm had some red rubber pieces. So and they're especially good to pick up very fragile rocks, or also especially made for corals to pick up such things. And now to break off the rock, we hope that it's going a bit easier if we use the pot arm here that doesn't have this red rubber pieces on for fragile work. So
So you're not gonna be able to do that trick I told you on this one. Just so that's something this is a vertical wall. So if you right there.
It's the worst I can do. And I wasn't.
He seems like
Pushing it pretty good.
very militant about.
Grab those in there.
Just trying to put that face down.
You gotta try to get your tooth and you gotta get your teeth.
Authoritarian.
At least one finger underneath it. Too high. I think you need a little bit. There, get that tooth in there.
The geophysicists would be that just it's hard to stand still. Usually you're surveying, you're going traverse. It's the stuff. Look around for the stuff.
I mean, if this material is actually in situ, it's flowing down the hill, then that
There could be
didn't
some
form at an
thought.
ocean bridge necessarily. Like it doesn't look like something that formed in this goes up or it's falling down.
Yeah.
So it could be good.
Try to get, let's see, with your port arc, can you grab, any weapons on that for that? Yeah, maybe right here where your right leader was.
John, can you show me your strategy?
Oh,
I'm just curious.
Because it's your homework. You tried to bet you are being great.
Yeah, where was it?
But it looks pretty.
Well, this would be a big guy.
How far
A
was
little big guy, about a baseball, softball.
So this is our first dive at the Kane Fracture Zone. So we arrived here in the area at, I believe it was yesterday afternoon. And we started out with making bathymetry maps for a few hours once we got here to understand the details of the seafloor a little better, and also for planning AUV dives and CQD tows.
Is that too small?
Melissa? Melissa?
Well, that's pretty. I'm happy. Can we take a IO or rock
So we got one.
Thank you.
Did it rotate?
Yes.
And then last night we did CTD tours. So to understand if we find some water column anomalies, like chemical and physical anomalies from active hydrothermal venting. And then earlier today we deployed the AUVs into the seawater, so to give us 1 meter resolution seafloor bathymetry maps, also to find interesting features that we can dive on later, or also do some chemical or CTD CASPER chemical analysis. And now this is our first dive in this region to just explore the geology, understand a little better what we are working with here, and the chance for hydrothermal vents, we don't have any indication that there is anything, but you never know. So we do have our fluid samplers on. We could sample if we would find something.
All right, moving forward. We got this spot?
We move up, yeah, we're good.
I've heard that before.
And then we were good.
I'm not going to be interested in another sample for at least another 190 meters. So around 1300, it's going to get steep again. And until then, we can kind of just go pretty quick.
I'm going to hold you to that.
J-Rod is not convinced.
You'll believe that when you see it.
Yeah.
There's going to be something.
Okay, let's move on. We are ready.
Speed up.
And then it's like stop.
Speed up until we want to slow down.
Speed up, slow down, speed up, slow down.
Okay,
That's all good.
moving on.
Look at that.
More. Oh, now okay. There's a break. So there are the pillars, and then the break off on the right on the side, with some sediment sitting in between the next little ridge of pillows. So we're still moving up this slope here. Whew. That's pretty impressive. Some of these pillows are still very much intact. That's great.
We're starting to bring you in flat or something.
Follow this route for a little bit.
Yeah.
You want to see that rock over there.
No, I think we're good. Oh wow, they are big.
Comes and goes.
Yeah. This sun. There's so much life here. That is impressive.
Can you just slow?
What
I just want to take a
was that?
picture, not slow. Okay. That looks like a scarp, huh?
It's our orientation. We're facing south.
Yeah. I was just trying to follow this while I'm going down that line.
So what's our heading
We're
right now?
facing northwest, 123.
I was facing southeast.
Southeast.
Okay, that's
You want me to follow this for a little bit?
A bit and then further up again.
You can just keep going uphill. That's cool.
I think up is.
I mean, this is kind of.
Then follow
If
it
it's
up.
It is up. It definitely is up. I'm not off the line yet.
We continue moving upwards along this scarp here. That's great. More sediment cover here. Still some intact pillars, but mostly talus.
A long way to go. Can you use your arm and get rid of that hose?
Until the very end.
So while we're doing this geology dive, looking at the structures and the geology, we also sample for biology. We do collecting rocks for later analysis and also some fluids. So some of the fluid chemistry we are doing here on board. So in our laboratories we have several instrumentation, or we have instrumentation set up. So one of them is a gas chromatograph, where we measure the methane and the hydrogen concentrations in diverse type of fluids, like some that we collect in the water column from our CTD class, and others that we collect during the dives in different sampling bottles. We do run pH. We also subsample for many other chemical analysis back on shore, so that we get the full composition of all the fluids. And we do also have the possibility to do gas chemistry. So we have gas-tight sampling bottles here that we then subsample here on board. So we separate the gas from the fluid phase and also prepare them for later analysis on shore. So from the samples we are collecting today, from all the fluid samples, we will run them for methane and hydrogen tomorrow morning to see what we find, and hope for more information of where to look further for active venting.
Between 80 and 70. And you don't have to just constantly do it. You can like twice a day. Probably Friday, and the SPs will call well, two breaks.
So now we have a little bit more sediment coverage here. We're still climbing up. We are now at 1450 meters. More sponges.
I'll be watching. Don't want to confuse the guests.
I fixed the autofocus on the cleaner. So it's
Oh, sure.
and it's in autofocus. That's what they make me make this.
You got this
People.
area now.
All right, so now I can talk about some rocks. My name is Melissa Anderson. I'm a geologist at the University of Toronto. And one of the things we're looking at here is basically going up this entire steep scarp and taking some rock samples along the way and seeing what the different horizons of rocks are made of. So along a steep slope like that, it's kind of an analog to drilling. If we take a sample at the very bottom and a sample higher up, then the sample at the very bottom is the oldest material. So we can actually look at the evolution of this entire large portion of the crust by sampling at different depths as we go up. Right now we're just seeing a lot of blocky material that we call talus. And this all looks like mafic volcanic rocks, but the really dark color is manganese oxide coating the outside of it. So it's really hard to know for sure what it is, but based on the environment, we expect it to be mafic volcanic rocks, and that means that it contains a lot of magnesium and iron. That's what mafic stands for. So dark colored, basically your basalts, or the kind of thing that you would find in a lava flow at Hawaii, for example. Some of the different rocks that we've seen a little bit earlier look like they were lava flows that were in place, and they have this really cool bulbous morphology. So these are what we call pillow flows. The bulbous things look like pillows. So what we really want to sample are things that look like they're in the same spot that they formed. So these little pieces of rocks, we don't know how far they have fallen down this slope as we've gone up. All right, there's a fish. You can tell the animals really like the rocks too, so we have that in common, the animals and me. We're in a little bit more of a gentle, well, it's still a steep slope, but it's not as vertical as earlier, and we're in about 25 or 30 meters, we're gonna hit another really steep slope, and we'll see if there's still a lot of biology on that. Yeah, a lot of biology on the bottom. So with these really steep cliff faces, there's a lot of currents that can move and upwell along the cliff face, so that's why a lot of these different creatures like to, what's happening? Like to hang out and make their homes on these walls of rock. So we see all different sizes of talus coming up, and some things like on the left-hand side that we see there, that looks like a bit of lava flow poking through. Some of the smaller blocks probably came from somewhere up above. Do you want to take over?
Take some more fluids.
Yay, we're gonna take more fluids.
Musical chairs in here.
Musical chairs.
Yeah. So we're just doing the same thing as before. We keep moving, and at the same time, we're gonna take some fluid samples. All right.
What sample number are we up to now?
You are up to number 16.
16. Okay. Ooh, that's cool. Nice shot.
Oh wow.
That's beautiful.
Is this going to be HFS?
That's correct. HFS, yes.
I don't suppose you want to take any gas tanks.
No. Yeah, so the gas tanks are specifically for very gassy, high temperature fluids. They're designed for that. So I typically don't get good samples from this type of environment with the gas tanks. So we're sticking to our other fluid samplers. Now, did you say which number did you say? Sorry. 16.
So we can
Alright.
take, well, whatever. We can take, we did bag 23. We can either take, bag 22 or 21. When you get to 19, that one's filtered. 17, 18, 19, or 15.
I think I take 22 then.
Okay.
Or do you want the filtered
20,
one?
22 is fine, yeah.
And then I go select.
Mm-hmm. Hopefully it's, oops, it's not happy. Need to, sometimes it falls asleep, so. Maybe that'll work. Yeah, that works.
So I'm still learning to use the fluid sampler, so Dave is giving me another lecture here.
So what happens is, if it hasn't had any commands for a really long time, it just goes into sleep mode.
Okay, so I wake it up here.
It happened twice.
Okay. How do I see that it is happy? Or
Well, it had a red light,
mm-hmm.
and it said when you gave it a
I'm not
command,
ready.
it said it's not ready because it was sleeping. So
Okay, so I tried it again. If I can select bottle number 22.
okay, now
Now it says
it's
no.
okay. Try it. Click 22 again.
Yeah. So it lost the information there.
There it goes.
Now it starts rotating. Yeah.
And we can tell it to take 450 mils.
Right it in, yes.
And somebody has to log it. So.
Can you help me?
Yes, I can. All right, HFS is starting now.
Oh yes, I'm ready, or
It's-
close to ready.
Tell me when you want to go.
So this one is not running right now. And
Right.
this one is.
If you just hit the sample button,
Yeah.
it will sample
Then it will start.
that amount.
Yeah. Okay, I'm starting now.
We've streamlined this tomorrow, so instead of having fluid sample start, then log it, and then fluid sample end, when it starts, we hit the button, and then we
To get the
have
timestamp.
an end time here.
Okay, sounds good. Yeah.
50 seconds. You read that. It does tell you here too. It's synchronous.
Oh yeah,
it's
it's synced. Okay. Yeah. So here both are left. Both
Yeah,
pumps are
which
running,
means they're
and
running.
I have now 117 milliliters. Do you need temperature? Yeah. I'll give it afterwards, I guess. It's gonna be back 22.
So B22 or filter bed?
Just unfiltered, just in 22.
You can put in here. There's unfiltered bag, and since we're moving, I've been putting the depth where we are.
Yeah, we are
so
1400.
So it's
Oh.
easy to put into our file. Of course, it'll be in the C-log file too. So we're just gonna. This is all repetitive, I suppose. At least we have it written down. Yeah, I've just been recording the depth interval. When it gets done, oh, that's almost there. Alright.
Was that 450?
1387.
Yeah. And we start, so we sample between 1400 and 1387, so over 13 meters. I do need to close, right? That's the next one, yeah. So close, I do, yes. So the maximum temperature was 4.7. The average temperature is also 4.7. The volume is 450. Same. Best. Yes.
On the anemone or on the yellow sponges?
Is when I push the close button, does that just change the valve, or what does it actually do?
Whenever you're ready.
Oh, okay.
Oh.
All right.
Perfect, thank you.
It's, it basically just marks it as done.
Okay,
So
with the break
doesn't
frame
really
and
do
like we
anything
used
else.
it up and yeah. Okay.
So that's good?
Yeah, perfect. You go ahead.
Do I hit in there?
Yeah.
Fish.
Let's see, we got
Do you want also filtered one or not really? Yeah.
just take one every once in a while.
That's kind of different over there, huh?
I don't know. Probably just taking one every couple hundred meters up the slope here.
Okay. Do you want to test the full sampler, like all of them again, or just however many we end up with?
Yeah.
Yeah.
If we just keep taking one like every half an hour or something like that. Or
Okay.
we have Niskin bottles too. Could take a Niskin.
Do you want one now? We'll put right on top.
I guess we could probably wait. It's like, there was one Niskin taken for eDNA.
Okay.
But that's it. That's all I've taken.
Okay.
Number one, just.
Do I know which ones are piston and which ones are back, or is it like I have to talk to cabin, or is that somewhere indicated in?
Yeah, you know, I didn't write it in there this time, but I can put it.
Yeah, so Dave is to give me information about fluid sampling so that we can use his sampler through over the next few hours and collect some more fluids every now and then so that we can run them in the morning for dissolved gases and also subsample for running other analysis back home in our labs. Okay, I see.
There's only 7 bags in this. No, it's 7 bags. So 24 is oxygen sensor. If you want to run that,
Oh.
switch to. I think we could do that right here.
Yeah, it's steep, but not as vertical as the sunshine.
I didn't know that there's an oxygen sensor on the fluid sampler.
Sometimes I have a pH sensor, but I don't have one.
Yeah.
And then
Okay,
just
so you
run the
just selected
pump,
it, got
just
the
run
right
it.
position, and then
This is the oxygen
Mhm.
line here. It's milliliters per liter. It's like a weird unit.
Yeah,
but that's what it
that's
is.
the oxygen unit, right?
That's anemone. That's an orange one.
So that's milliliter per liter. And then I can just also close and that ends the oxygen
Actually,
reading, or just go to another position, or
usually just let it pump for a while,
Mhm.
and then just you don't have to push close, just push stop. Or hit
like
the
sample
if you
again,
hit
that would
again,
stop it.
turns
Okay,
off.
yeah.
So you can write more or less whatever you want in there. You don't have to write anything for oxygen. But,
Mhm.
looks like it didn't change. Well, maybe it's going down a little
Yeah, I
bit.
don't know.
Swimming backwards.
Yeah, that's an. Maybe it's trying to sneak away from us.
Doesn't work.
Doesn't work. Yeah, you.
It's better at swimming backwards than this camera as an autofocus. That won't.
How big is that fish?
Just over 10 centimeters long.
It's a sign of the sea. They're all just piled up there. Sponge graveyard.
How far away are we from the summit now?
300 meters.
300.
Well, 350. It's about 1,000.
I show 428.
428. Okay. Yeah. Nice. Thanks.
To the top of the scarp.
Yeah. So we still have some ways to go till we reach the summit.
How fast you want me to go?
That's fine. No, you could. We still want to see something. But there's less life, what it looks like, and well, there's still some.
There used to be like,
Yeah, yes.
looks like, has fallen down.
It did come down a little bit, the oxygen, yeah.
Could be just changing with depth.
Yeah.
We're coming up on this part of the slope. It's gonna start at about 1,300 and be steep.
Steeper than this.
1,100 steeper than this.
Oh wow, so
Steeper than this almost means like over.
It'll be pretty, it might be over.
we are approaching a very steep slope here for the next 200 meters. Okay. And then also, should I go to the home position after I finish sample? Is that easiest
Usually,
to
yeah,
start over again?
it's
Yeah. Yeah.
If it happens to go to sleep or you have to turn the power off, it wants to find home port right away.
Yeah. Oh, it says now the sample is not ready, so do I have to wake it up again? Oh no, it's moving. Never mind.
Oh, yeah. If you do that, bring it over when it starts to work. It's kind of weird, the messaging on there, but
Leave it off for now.
so now it's got to zero.
Yeah, yeah. So it just was like telling me I'm doing something you can't, just in case I wanted to do something else. Basically telling me it's working.
Well, yeah.
Yeah.
If it's in the process of doing something and you push any button that's
Yeah,
a command,
it's like,
it'll
yeah.
say no.
All right.
It's a yellow stuff.
What's that?
Yeah.
Bright yellow.
Well, we're higher up, so maybe it's a nutrient well.
We still have some pillows here, but most is just tailless.
Do you want to zoom on any of those?
The yellow ones we could.
Yeah, those bright yellows.
Do you want to? Yeah. So
On the top left there, zoom on that rock.
Own it.
Those are nice.
do you know what it is, John?
Weird
Yeah.
sponge.
A sponge, yeah.
Yellow sponge.
What is nice is that now I do not see the sponge spider that were not common. So could be a kind of solution.
Yeah, it seems like it.
Yeah, we are looking at different type of sponge here than we have been seeing before.
That good?
Yeah, perfect. Thank you very much.
Thank you.
There's another big outcrop over here. If you want to, like, some bigger ones. A bigger version of that yellow is right. Yellow.
I think J-Rod enjoys his job a lot.
Yeah.
Oh, look at that pillow in the middle of that screen.
Yeah,
That's a
it's a nice
nice
one, yes.
round pillow.
I'm looking at yellow sponges right now.
No pillows. Yeah.
Did you want to look at
All right.
some pillow or keep going?
No, I think
Zoom out.
we're good because the pillow is right here in front of us. It's coming up. Yeah, yes, the big guy here just in front of us. Yeah.
That's a perfect pillow.
Yeah, this is really, really nice one. Yeah.
You and me sleep on different pillows.
Yeah, these are
Yes.
rather uncomfortable.
All right. Yeah. Then just moving on.
Be somebody home. Have y'all looked at any of the shrimp inside the sponges? Yeah. Have y'all had a look inside the sponges yet?
Inside the
I don't
sponges?
know. We
Oh, there's
can.
something in there.
Yeah.
Peek in there.
Can you check it? Let me get set up.
Want to look inside. Oh,
Yeah. I can look at that one and then we'll see if that one.
We're checking out the inside of a sponge now.
this is really cool.
It's coming up. Yeah.
I like
Nice.
But this is cool.
There's somebody home in the other one. Let me go to the other one.
Was there someone living in there in the other one?
Yeah.
Yeah.
There's somebody probably living in that one too. Just
Yeah.
the other one. I'm pretty sure I saw.
Ah, here we go, the big one.
Pretty sure I saw somebody.
Yeah.
It's deep and hiding
Yeah.
now.
There might be some. Yeah. Oh, nice. Yeah, there. It's definitely hiding. There's someone else further down. Yes, yes.
Oh, sorry.
That's all right.
You can't do it with that one. With you have to do it with what I have on here. But it's kind of hard to do when you have.
This isn't too uncomfortable sitting here on the stool. It's all right. It's all right.
Nice. Well, that was a nice excursion into the sponge.
If we get one of those wedding sponges, it'll be better.
Well, that was quite cool already.
Never worked for some reason.
Yeah, it's a very different sponge community
Yeah.
here, isn't it? All the old
Yeah, definitely some zonation going on with the fauna.
I really think so.
Yeah.
Species of corals haven't changed that much in sea.
Yeah.
Some of them, but the community of the sponges have totally changed.
Yeah, so Juan is saying that there's a big change in the community of the sponges, but not so much in corals. They're more like this. From the corals, we see the same ones still, but definitely not from the sponges.
I'm not sure to tell whether or not those rocks are mostly in situ, like in situ or
I think some of it look like in situ, but there's also a lot of like just yes, tailors mixed in.
oh, we're back to the like workings.
And now we have that's definitely an outcrop here.
Starting to get to vertical land.
Yeah, it's very steep here, right? Yeah.
It's getting more though.
Yeah.
Oh, 25 meters. Oh, that's cool.
Oh wow. Climbing up the wall here.
Well, people were talking about wanting to log if there's any like debris or garbage. I would see it.
Oh. Yesterday we saw a can.
Oh, you saw
Yeah,
it?
yeah.
Okay.
Otherwise nothing, yeah.
That's good.
We are logging.
Yeah, I think it's like human something.
Anthropogenic
Okay, yeah.
observation.
Pretty clean and undisturbed place.
Yeah. That actually looks like mostly in place, even though it's a bit fractured. But yeah, it looks like it's all still in place and not broken off pieces of lava. And we still have the yellow sponges climbing up a very steep slope here. Yeah, with the
Yeah,
the dark coating on the rock. So this is a manganese crust. What is the red one here?
Sea urchin.
A sea urchin. Okay.
Are you sure about that?
Yeah, it looks like one of the.
Oh, oh my God, the jellyfish.
Get that guy.
Ah,
Oh,
oh wow.
nice.
He was just hanging out.
Mm-hmm.
Yeah.
Not anymore.
Got disturbed.
Yeah. Just taking a rest when we disturb his nap.
Still this,
That's nice shrimp.
That's a big old.
yeah, big one, big shrimp, and another one right up there.
We're pretty close to 1,300 meters. If you guys want to look at it, get a rock, then you can do that here.
You want a rock sample?
Between here and I said I wouldn't take a rock sample before 13.
So you want to
Close, and there's cool,
plus minus 15 meters.
Yeah. I mean, just to see what's what.
You want to set up then for rock, or rather wait a little?
Yeah, let's set up.
Okay.
Just might want to take the time.
Okay, then we're gonna take a rock sample here.
Roger that. Any particular type? Size?
Can you zoom out and then I can see?
Sure can.
Yeah, once you do that.
So in this area, we just try to collect our samples nicely spaced so that we get some information for rocks and fluids just over like the whole area. So we take them.
Close to here, so not like down here, but anywhere, any of these rocks in here would be fine.
Yeah. So we take samples every few hundred meters.
What about this one that's coming up middle screen right now? About to put the lasers on it?
Oh, you're.
Yeah, what do you think about that guy?
Yeah, that guy looks good.
Kind of looks like maybe they might come loose.
Yeah, sure. Or even like one of these guys.
Okay, I'll set up for that area. Port on again, so. If possible, yes. Good with that? You're gonna pour it on, correct? Try to get it center straight.
I'm wondering, I think it could be a good idea to close any skin here also because we have the different sponge community here.
Yeah.
So after we have the rock, I think we should close. Yeah, that's what I'm thinking. I mean, I don't know if we actually do see the difference, but it could be interesting.
Yeah, we should do that. I hope Sean wants another sample.
Oh, he doesn't know, huh? Shall I check with him? I can go check with him.
I can ask him.
Or that. Or otherwise, I just run it from methane.
Yeah, that's true. You do nothing with it if you don't want it.
So you're using less pressure.
So we are setting up for rock sampling right now. So it's not easy here because they are all in place. In place, that means these rocks are not just lying around. They're actually still in situ. So it's a bit harder to get them broken off. So the pilots are discussing what the best approach is, the best angle of getting a rock sample. And we are again using the port manipulator, so that's the one that doesn't have the coral cutter on it. So these rocks are not fragile at all. They are like the opposite. So we are using a bit more the less delicate sampler here.
So you look down You have to hire someone so that you can see what's on. You won't fly with that. It'll be like this. Good. All right. I'll be right back. Just come out screaming if you need them.
Hello.
Hello.
So Jared and Doug are exchanging pilot seats.
No, he's stuck on there now. It looks like the rocks are loose, but they're really easy.
Yeah. Yeah, so this is not an easy task here.
What about this guy down here? Oh, you got it. I take it back. You are. Oh, I thought that it was broken.
It's just from wiggling a bit, yeah.
Oh,
A teeny piece, but I don't think you want that.
no, thank you. The rock doesn't have to have fire on it.
What about this guy right here?
Do you guys want the sample that's finished? No. Okay, yeah, anything in there's good.
Sean says yes to a Niskin.
Yes, okay. Yeah, we're just checking with Sean, who is working with eDNA samples for here, if he would be interested in a sample. So he says yes.
Yay.
Oh, that's a little one.
I'm okay with the little.
Big enough? Okay, that's good.
I don't know the scale actually. Yeah, we'll take it.
Watch out.
Can we put it in the quiver actually? We'll fit.
Yeah, but it's open. I don't have anything that's open.
Can we just hold it right there on that side for 1 second? Totally upstairs.
Yeah.
Mm-hmm.
Very nice. Well, I guess it's about time for me to go to bed. I'm going to bed. So Kevin can do
Yeah,
this.
I'll
if you
ask
want
him.
to.
Yeah.
Yeah.
I will ask him as soon as
Be hard to get.
theory.
If you're going to keep sitting here, though,
We will exchange at some point.
hard for him.
Yes, please, in a quiver, please. If it fits.
I suppose he can sit here.
Oh, you would have had to open it first.
Or
Okay,
maybe we can move it over here. Let's see.
Rock box. 1A.
1A?
1A, please.
Oh, we might be able to get it in the quiver if you really wanted to.
No.
1A, all right. And you, watch out. Yep. So you want a Niskin here?
Yes, correct. Now we are setting up to collect the Niskin sample. Niskin samples is just the water samples from here. That would be great, yes. What is the sample number for this one?
18. Niskin 18, and it will be N2.
Thank you. Yes, Niskin 2. 13, 14, 4, 0.
Once they start moving again, could take another sample.
Yeah. Where do I see the flags? We have to go check.
Did it work?
Hold on.
I don't see it. Did it work out? Yeah, I can't see the flag, unless it got stuck somewhere. So the first one is closed for sure. Actually, it's closed. I think I can see it now behind the camera there. Yeah. Okay. Great. Yeah, then we can move on.
Oh, we did it? Yeah, you got it.
And so as soon as we are moving, I'm starting another one here. Yeah.
Started at 1,300 meters or something. We're almost there, I guess.
Yeah, right now we are at 13, 14.
Okay.
So you're right, then kind of brushing this.
So yeah, I started at 1,300. The last one was at 1,400. Okay, so we finished sampling, so now we start moving again, moving upwards towards the summit, and it's still very steep here. And while we are moving up, we will also take a water sample, but for that we are not gonna stop, we just continue forward. Should I have stopped something here, or they stop by themselves?
Right now should be the length to selecting should be going to home port.
I didn't.
What's that?
402. It's at this point. I don't know. To what it's up to. You were going from.
I haven't pushed anything
You weren't
yet.
even sampling.
No.
It was on home port already.
Yes,
So-
yeah.
No.
So I just wanted to start over now, and I was looking, and was like, I don't know.
To try to wake up again. Yeah. I guess it just went to sleep again. It doesn't really like to stay on for a long time without doing anything. Say already your home port double it.
Interesting.
Where do you want to go there? Piston?
Yes,
Getting piston
a
three or
piston.
We got
And
quite a few samplers left, so
okay.
probably try to fire all of them, I guess.
Okay. So getting in position. So the fluid sampling is still kind of slow because I'm still learning. So so goes. So we're still having. Yeah, so we still looks like we have the
anything here except the sponges.
the yellow sponge, and we still have some crusts. Looks still some manganese. I think it's very dark. Like
Compared to the number of animals that were present in the lower part.
I think there are a few corals also. Melissa, I'm setting up for another HFS sample. Yep. Let me know when you're ready, please. Yeah. Oh, okay. Thanks. And that's sample 19, correct? Yeah. Yep. Oh wow, beautiful. Yeah.
All of this will be recorded, just whether or not you want to add.
Yeah, when I say feature, I say feature with something, so I can remember what. Yeah.
So, Kevin, do you want me to have him come in here to help you with it right away? Or? He could, he's here.
Yeah, probably
Yeah.
it's easiest.
Okay.
Yeah.
You can explain it to him. I mean, he knows how this. I'll go talk to him. We're basically taking a sample every half an hour or so, or if we're going up and down like every 100 meters, and probably try to fill up all the samplers so we can test them as well as get some data.
Makes sense. Okay, Melissa, I'm getting ready here with the fluid samples. Yeah, I'll let you know. Thanks. So that's now piston, so it's 700.
Yeah.
19? Yes, thank you. Then
You can
anytime.
put it anytime you want.
They're all unfiltered, right? The pits.
I'm going to have to verify that from Kevin who set them up.
Oh, I'm not writing anything. And at 1300. So now it's ready, right? Yeah. Okay, I'm starting the sample. That is a piston number 3. We have to verify that first, so just don't. Yeah.
I'll go ask Kevin that now.
Okay, thank you.
I'll ask him to come on in. That's what? Yeah. Yeah. Yes, very steep.
Yeah, we are in an extremely steep wall here. Still, lots of the orange sponges. Some other ones as well now. And while we are moving upward, we are collecting fluid. It's almost a vertical wall here. We still have the yellow sponge that is dominating, but it's a little more diverse again here. Okay, the fluid sample ended.
Okay, and can you tell the time is
4:14.
4:14. Okay, and was it
Then
filtered piston or unfiltered?
we don't know yet, so we leave that open. Then we have the maximum temperature is 5.0, average is 4.9, volume is 700. Skip the second volume, and I think that's it. The depth is 1300 to 1288 meters in additional text. 1300 to 1288. Thank you.
So should I just call it P3? Like I have
P3 is fine. You have to use one. That unfiltered is more likely. Yeah. We will check afterwards. Thank you.
This is so cool.
Yep. Okay, then we can move. Yeah. No, we were looking at this coral, but yeah. So then we move on to the summit still, and the next sample will be at around 1210 meters.
Doing beauty talks here.
fair enough. Look at how many stars are on that thing. 5 of them? How vertical that wall is.
I am now.
I don't know what all of it is. So like one of those individual things.
So we did some beauty shots, so now we are moving on upwards.
If we can get a zoom in on individual columns.
Oh, okay. So we're gonna zoom a little bit more. Super zoom, yeah. So Monica, do you want to? Yeah.
As close as you can get to that, because we need thank you so much.
You get another meter till you're on it.
It's like one. Is that okay, or do you want to take it closer?
Okay, now we are zooming in on the individual branches. I don't know what the word is for this.
Yeah. I just didn't know if she meant this thing.
I don't know. Maybe where to move it. We are right now at 1283, and the next fluid sample will be around 1200.
Oh, okay.
Yeah, so we still have a bit of time here. Yeah, it's kind of we can rearrange things afterwards a bit. Also, I'll switch with Melissa at some point anyway. Yeah.
Switch chairs. Do you want to switch spots? I'm happy to move over there if it's more comfortable
Yeah.
for 2 of you to sit here. Great job, thanks guys.
You happy?
Did we get an image?
If I unplug them.
No, we haven't found anything.
No, I mean, unplug if I move it.
Aha, we can set it up right here.
Let's switch spots.
She wants the lasers on it, so it will be
Musical chairs, musical chairs.
So Melissa is coming on now.
Alright. It's these. I think I got it, guys. Can I thank you very much. Thank you. Tomorrow I need to get the chair in here, so if you want to maybe take the chair and try to show.
I'm not going to sit here. I'm going to sit with the methane spectrometer.
Okay, I just needed to get in here. Yeah, you can carry on. Thank you. Ooh, some tall person was sitting in this chair.
So they just took some additions and this right here. Yes, here it's more familiar.
Here's my chair adjuster. I feel like a child. All right, let's keep going up this wall. We're getting pretty close to the top. We have another almost 300 meters to go. Sorry, can you pan to the right just so I can look at that slope again? And just kind of fly with the far off image in the back too. Yeah, that would be great. Oh, that really gives you a sense of how steep it is compared to looking directly at it. So mostly what we're seeing are lava flows that happen on the surface rather than exposing the deeper, what we call intrusive rocks. So a lot of these flows are pretty in situ, meaning that that's what we're seeing is where they formed. That's so cool. We're coming up on a depth of 1270 meters below sea level. We started at a depth of about 2,000 meters, so we have come up just over 700 meters from the start of this dive, and this is the steepest part. Are you okay? Sorry.
What's the number to the bridge?
I mean, we're not the feet, right? But I'd say we should probably, if you want to be careful, keep bottom by
4:30.
I'm gonna double check with zero when he gets back, but I'd like to leave a little extra just to get all the floats off. Yeah. 30 minutes, so we're at 1,000 meters.
So we're provisionally calling this area Sponge Heaven, which is different than the earlier Sponge Heaven that we saw, where we had all of the light-colored sponges, and here we have all the yellow ones. So these steep walls are really great for our deep currents to upwell along these faces and bringing nutrients with it for all of these filter feeders.
So it's 4:30 off bottom.
4:30 off bottom, yep. We'll be at the top.
Here, it's great technology. What's the transit time from our location to where we are typically
Coming off the bottom.
So
coming off the bottom.
All right. Okay, so is it at the end is closer to the pickup location then? Okay.
So that right there in the middle of the screen that's kind of rounded, that's what we would call a pillow flow. And the outside of it is all broken up, but still essentially intact. We can just keep moving up, we don't need to stop for a while. Unless we see something really cool.
Thank you. So it's half an hour to the Peru area in the front end of the circle. And that's if you take how far we'll get on the map.
So somewhere I am now.
Yeah, I'll say 30.
Can we ascend a little bit quicker? Yeah, just to cover more ground. This is pretty samey.
Turbo.
Turbo mode, engage.
We're okay with 4:30 out the bottom.
About 1,000 meters. I can tell you exactly. When we get to the top of this scarp, it will be. Actually, it says 1,075 meters at the top of the scarp, and then it's pretty flat to waypoint 1 at the top of the scarp.
What about the one that says summit?
Yeah, that's like a little bit further back from the scarp. It's at the same depth of 1270.
So top of scarp.
Yeah.
Actually the highest point.
Yeah. Oh wow, look at this.
Wow.
So cool. Oh, and even more sponges, lots of dead ones too. Yeah, another 40 meters or so, and then we can try to pick up a rock.
Pretty big overview.
Yeah. I mean, if something like that you see looks easy to sample, then we can stop, but getting a little bit less steep with all of the sediment. We've come up to 1230 meters below sea level. Just a huge amount of volcanic lava flows covered in this, these yellow sponges. So the higher we go, so we at the bottom of the cliff, we're looking really far back in time when we look at the rocks. And we're kind of slowly moving forward in time as we go from the bottom to the top of this cliff. So it's really, oh, did you just? Right. On the map, it almost looks like kind of a ridge that's coming out that we're going up as well. So an old dive reported seeing some evidence of old hydrothermal activity near the top of this scarp. So maybe at some point there were some black smoker chimneys where the fluids moved through the rocks and caused some alteration of the chemistry and the mineralogy of the rocks. And so if we see something like that, we'll be quite excited and we'll stop and sample as well.
Yeah, I've been working my way that way. Still trying to keep the features. Like the lesser corals have been knocked through.
Lots of small talus right here. Okay, what kind of sample?
Filtered bag.
So HFS. Okay, filtered bag and the bag number.
18.
So your sample number is 492 HFS 20-FB18.
It's just what I would do. Just happened, I'm sure, many times. I'm going to take a piston sample too. Wow.
Okay, so that will be 21, and do you know your piston number? And if it's filtered or unfiltered?
Unfiltered, that'll be piston 4.
Okay. Let me know when you start taking that sample. Some point around here we'll do a rock sample, but we'll finish the fluid samples first. Yeah.
I see a lot that have that texture on them. You want one of those?
It doesn't matter what the texture is for it. It's any rock that looks like it is sampleable will make me happy. Did you already start the sample?
Oh yeah.
Yes.
I started at 432.
Yeah. So,
It's 300 mils right now. Of the 700.
when it ends, also just let me know so I can make it over time.
You can maybe alter distance in bags. Yeah, we plan on taking piston 4 right after this. Okay. Alright, I'm heading down. When's filtered?
So we have 120 meters to go to the top, and I definitely want to get a sample before we get there, so we can also be a little bit slow here.
I'm heading down, Melissa. Thanks.
We'll see if it gets more exciting at the top.
I'm afraid I'm gonna miss the top. Gonna get some sleep and get up early.
Sounds good.
Okay.
So right now we are taking a sample of fluids. We're going to look at things like the concentrations of, what are we looking at?
Methane, pH, trace metals.
Methane, pH, trace metals. They've taken a few samples of the fluid as we're going up. We've also been sampling the rocks to see the changes in chemistry of the rocks over time. I'm kind of surprised to have seen so many lava flows along this surface rather than intrusive rocks, rocks that form below the surface that are now exposed along this steep cliff. So a lot of these were kind of going down the cliff. Yeah, I think we're almost getting out of the lava flows around here, so if we can just go kind of slow.
Sample stopped.
Sample stopped, okay, and let's see the time. 0436. And the sample depth range, did you get that?
1210 to 1180.
80. And the maximum temperature?
That's 5.4.
5.4, and the average?
5.4.
And the pump volume?
635 mils.
635. Did you need a best volume?
What?
Do you need a best pump volume? Or just one?
That's good. This is sample.
Do you mind if I take a rock sample before your next fluid?
Sure, go ahead.
Can we pick up one of these rocks? They all look kind of loose. Yeah.
Shortly of the looseness, and there's kind of like the left area. Well, I'm tell that last time I was trying to look for.
I think a lot of these should be pretty loose. They don't look like they're totally in situ. It's just whether or not they're kind of cemented in place.
Okay, I'm settled. Scrape on the wrist. Okay. Look at that scrape on the wrist. That's a mark. Yeah, it's not a ramp. Okay. Let's try it. I'm just gonna poke a couple and see how I move it all.
Mm-hmm. We do some very scientific rock poking now. The ROV team is seeing which of these rocks they can actually get a hold of. They're surprisingly stuck in place, even though they're broken fragments. They're kind of solidified in the rock. Ooh, nice. Beautiful. There's a little bit of sponge stuck to it, so the biologist will be happy with this sample too. Ooh, good one.
Where do we want to put this?
In rock box, what's the number? 1B. And I would like to take a couple pictures before it goes in the box, so that we can tell the difference between all of the rocks that we've collected. Okay, so just a picture like that. We don't need to zoom in too much. Just a little bit, not too much. That's perfect.
All right.
And then 180 degrees on the other side, please. Perfect. And into the box. That's great.
Which one again?
We are looking at rock box 1B, so
1B.
on the top right. Yeah.
So we over by the Asian Festival. Yes. But we'll retract the. Anyway.
Ooh, it's pretty big.
Beauty.
Nice. Thank you. All right. We are good to continue going up.
We'll be on top of the biolabs. Yeah. Still on top of it.
Okay, let me know whenever you want to start the next fluid sample. We'll take off now.
Happy?
Yeah. Okay, just tell me when you hit go. So this sample will be HFS 22P14. It's going to be 22 because 21 will be the rock sample. So it'll be 22. And then P4 because it's an unfiltered piston, right?
Yes.
Okay. All right.
Oh yeah, I was going to. Sorry, I thought you sit down.
Okay, I started the distance.
Started the piston sample. All right.
Moving on.
Moving on up. We are getting really close to the summit now. We are at 1175 meters below sea level. We're probably getting to kind of a flat area first. So now this looks a lot flatter, yeah. And I think it'll go flat or flatter for a little bit and then a little bit more steep and then we'll be on top.
So just step up.
Yeah. Alright, so we continue our transect up this really steep, almost vertical wall, and we're seeing a lot of different kinds of volcanic rocks. That's all the dark stuff that you see. Some of the volcanic rocks look like they are lava flows that happened right here. We call that in situ, and some of the rocks look like pieces that have fallen from above. These pieces or blocks of the rocks we call talus. So that big jutting out piece that we're flying by right now, that looks like it is an in situ flow. And this kind of lumpy, bulbous shape is what we call a pillow flow. So as this lava is moving, the outside of the outside of that lava cools really quickly because it's touching the cold water, and it forms basically a glass crust around the whole outside of this little flow. It's like a tiny little lava tube. And the way that these things move, it really looks like a tube of toothpaste. Oh, that's interesting, Ross. We've got some more pillow flows, getting a bit more gentle the slope. You can kind of tell how steep the slope is by how much sediment you see on it as well. And the rocks look really dark because there's a layer of pretty thin manganese oxide on the outside of it. Alright, so this is another area where if you look at the bottom, you can see some sediments, but you also see this kind of crust of manganese oxide as well. Oh, 1 second. Let me just make a note here. Manganese oxide. Slope. Rock with a coral?
Yeah,
Hold on. I need to know what kind of coral. I don't know. Hold on 1 second. What time? That is 4:49.
Yes.
And the depth range?
1180 to 1140.
And the maximum temperature? 5.6. And the average temperature?
5.4.
And the volume?
688 pounds.
Okie dokie. Are you going to take any other fluid samples?
Yeah, probably at 1100.
Alright, we are seeing a lot of sediment and a lot of manganese crust. That's the kind of layers that you're seeing. And it looks like that sediment is pretty much all biological in origin. The light colors, and then you see these dark, like spicules. So this is a much flatter spot. Yes. Okay, can we zoom on the sediment, please? We want to take a closer look and see what kind of biological debris is here. Okay, what would be good is if you see something that you want, then come and yeah, I'll keep my eye out. We only have one more area where it's pretty steep, so that'll be the last one. Okay, sounds good. Alright, our biologists are going to be discussing what they're seeing and trying to figure out what all of this debris is made out of. They look like little tubes, some of them, don't they? Do you guys want a scoop of it to look at?
No, that's okay. I don't know. Yeah. Thank you though. Yeah, yeah. If you want to continue on, that's.
It's okay if you want to take a look. We're now at a depth of 1135 meters below sea level. We've just gotten to an area where it's a little bit flatter, where there's a lot more sediment.
Carry on.
Carry on.
Very nice.
Oh, John says he does want a section set for it. Sorry.
All right.
Oh, up here? Okay.
Yeah. Just of the sediment, right?
Yes, I believe so.
So that will be sample number bio 23.
Do you all have a preference which section sample or jar we put in?
No, whichever one works would be good.
That would not work.
No mesh.
Yeah, not the ultrafine masters.
Or no mesh at
No
all.
mesh would be better, probably, right?
That's fine.
If you want to get those spicules.
That's fine. We only have one with no mesh though.
I don't think.
We have fine, super fine, and then no mesh.
I think probably the.
The black things will make you fine if you want to see what those spicules are.
It doesn't go through the mesh. Like you're not going to hold it on the other side.
Oh, okay. Fine mesh is good.
Okay. So I think we're on a fine mesh now too, right?
Suction sample 2.
So. Okay. You do?
Alright, so the ROV is parked and they're getting ready to grab an arm that has a little vacuum on it, and they're going to suck up some of this sediment. And then the biologists are going to be able to take a closer look at all the material that's in it. We did get, if you look right there, you can see that one of our rock samples has a lot of this yellow sponge on it as well, but we still want to know, right?
I think we still want one that might take some of that sponge on it.
Okay.
Yeah.
So Chris, are we looking at this right? You're thinking that we're in two? Okay.
Yeah.
And two is fine mesh, which is they're okay with fine mesh. So we should be able to do that, right? Yeah. So you should be a go for section. On.
Are we on bottom 1?
We're on 2.
We're on 2.
Woohoo! Vacuum time! Clean it up.
Working good now.
Yeah, nice. So much better.
That's pretty cool.
How much of this y'all want?
Fill it up.
There's plenty down there, I don't think.
Are you my friend?
Some like shelly looking fragments, maybe you can suck those up too, just a little bit down. Yeah, there you go. Some of the yellow. Yeah, there you go. They're probably good with that. Give it a shake. Get in, get in. All right. I'm happy. I think they're happy. We got quite a lot.
Yeah, I think so. Not refined, so I think we're good.
Okay.
I see the jar is now opaque.
Okay.
Yes, so
So we're gonna pass through this flat area, and then there's the final. Coming off bottom a little. Question for bio, do you want to keep the rock as well, or you just want to take the bio off of it? Or keep rock, so we'll call it a bio sample. Okay, plan for the last little bit. We're going to traverse this rather flat area and get to our final vertical wall. On that wall, we want to take a sample of a rock with some biology on it, either a fan coral or a sponge, and another rock that doesn't matter if there's biology on it. So just two samples, and you'll have to tell me if you want a fluid sample.
Yeah, I will.
Maybe we can try to do a rock sample first, and then a fluid sample. Just take a rock as soon as you see a good one, and then you have the rest of the time where natural light.
Where's the summit?
We're almost at the summit, so we are in this flat little thing right here, and then it's just up a little bit. So we're getting to 1,070 meters-ish, and we're at 1134, so
My next one. Yes.
okay, sounds good. So we can go pretty quick over the flat stuff too.
See how here it says total 14. So if you go up to 15 per plot, it will be one rotation.
It's now 2 a.m. on the ship. We're approaching 1,130 meters below sea level. We started at about 2,000 meters below sea level, so we've come up quite a lot. And we're almost going to be at the summit over here. We should have one more beautiful rock face, if I'm lucky, that we can collect another rock at. Right now it's just a lot of biological debris all over the seafloor. Oh, there we go. That's cool. And we start to see some pieces of rocks, so that means we're coming up on that vertical face ahead of us. Alright, we're gonna get rocky here. And then we're gonna go vertical. No, so all these rocks, we don't know really where they came from. They could have fallen from above. So I would like to go somewhere where we can see these pillows and try to sample near that, so at least we can have better constraint on where it formed. But before too long, I'd like to pick up a rock when we see it. I've just learned that I can open 2 logs at the same time, so you can fire at any time, but we will have to stop for the geology sample, so if that makes a difference, we should just try to
What place geology stuff?
whenever we see a good spot.
Oh, okay, so so possibility I might fire one off before that.
Yeah, but if it matters whether or not we're stopped, then that could make a difference. Okay, yeah, good then. So we now have a steeper slope and more small scattered talus, and the substrate looks like it's a crust of manganese oxide. So what we're looking for is a nice lava flow to sample if we get lucky. Or a rock with some biology on it. What kind of sponge do you want? One of the yellow ones?
That would be okay, yeah.
Okay, starting
the sample.
Start the sample. Okay. And what kind of sample will it be?
This 5
Filtered or unfiltered?
unfiltered.
Okay. So your sample number is S0492 HFS 24-P5. Okay, there's quite a lot of biology on these rocks, so maybe at some point we can grab one of the, like that guy in the middle there that looks like it has a few sponges on it. Are you okay with that? Or you want to wait for the good stuff?
I mean, if you think there will be better stuff, maybe we could wait.
We don't have too far before the summit, so it might be that this is all we see.
Okay, so could we take one like something like this that has both this white and the yellow sponge, or like two different sort of morphologies, something like that?
If you see anything. It's going to be rock box 1C. Your sample number will be bio 25 dash rock.
Down there in the center.
Alright, so we're trying to collect a piece of rock that has a couple of different kinds of sponges on them for the biologists. Yeah, it's kind of hard to tell. Maybe right in the middle with that knobby thing poking out. But it doesn't... I mean, it has to be attached to something, but it's hard to tell what. Maybe poke around in there and see if there's a rock. Oh yeah, that one has both.
Yeah, because it has both the sponge and the, yeah, we've seen for that.
So it still might be kind of cemented in place, so it might not want to come up off the ground, but we'll cross our fingers here. Okay, sample in time is 509.
Oh, that's 1110 meters.
Uh-huh.
And temperature 5.8, average 5.8.
Sorry, what was the depth difference? Was it just 11 and 10?
Yeah, it's all at 11.
And the volume?
700.
Okay. Noted. See, they're all pretty stuck in place, so that black crusty stuff around it, this manganese oxide, is kind of acting as a cement for all these rocks. Oh no! The rock's breaking. Ooh! Don't think we're getting that one. Alright, any others? I mean, we might find a better sample location, but... Yeah, this is a bio rock, not a rock for me. Something with yellow and white is the request. No, we want the rock too. It could be that these are all too cemented. We might find something better a little bit further forward, so let's see. Yeah, let's move forward a little bit and just keep our eyes open for something that's not looking as cemented as this.
I don't know for sure, but I kind of feel like the flatter it is, the more loose ones there.
So here, my impression is this black crust. That's manganese oxide, and there's something like cementing all of the rocks together. So these should be loose, but they're not. So we can just keep our eyes open. If we get more vertical, then there might be that transition where it's coming up. We'll see. We'll find a rock with some sponges. Alright. These rocks haven't moved in a long time, I think. Coming up off bottom. Let's see if it gets less coherent. It's a lot of small blocky talents. Any of those rocks, maybe? I don't know. I don't know, we can keep going. Maybe just straight ahead. Looking for the... Which one do you mean? The white thing?
Or the stocky.
Sponges.
The stocky white one up there, or that one would be okay. With the rock, if you can.
Might be growing up between the rocks, so I don't know if we can get that one with the rock, but we can see.
That one in the upper left, it does look like it's on the block.
That one is.
Which one? This on the other top left? I don't know if it's loose. But if you can snip it, if it's not, that's what it's not. So if the rock stack is still on it.
Yes.
So you want to get the stocked one,
Yeah.
even if we can't get a rock with it?
Yes.
Okay.
Hopefully we could get a rock too, but if we can't,
We can do both.
okay.
Oh, look at the crab. Do you see the crab? Hello.
It's like kind of mean mugging.
Wonder how confused he is. Alright, so we're getting ready to sample this stalked sponge ahead of us. We are being supervised by a crab.
Yes. Everyone just has stock something or the other. And then we want one rock. We're looking to get 2 for 1, but we might just get the rocks.
I think that will fit into one of the quivers.
And we can share the sample if you guys want. Separate for quivers. Great.
So it looks like all the quarters are open except for starboard one. Open meeting.
So maybe we want to open, yeah. If we don't pick up a rock here, you can remove the dash rock.
Oh, no.
Okay.
Or what? your ROV. You can go stop it there, please. Too far away. Read your ROV. You can also take a bag right here.
Okay. One second. There we go. All right. Have you? How many you start?
517.
Okay. So your sample number will be HFS 26. Is it a filtered bag or unfiltered bag?
Unfiltered.
And the number?
It's 19.
Okay, so B19 at the end there. This is, sorry, section sampler 2 is the one that we have one in already, isn't it? Or not section sampler, oh yeah.
According to my map, it's one.
Okay, yeah, I confused, it's SQ2, yeah, that's good. Yeah.
Sample stopped.
Okay. The time is 5:20. And the depth 1100. And your temperature maximum
5.8.
and average
5.7.
and volume
406 miles.
Hello. Looks like we're picking a flower. I'm not so sure about this.
See the rock that's on back there? The back.
The rock right below it looks like it has some good yellow sponge on it too. That might be a good one to grab while we're here. So we're just repositioning now so that we can grab this stock sponge. We're a little bit too far away. The crab is still supervising, saying hello. It's probably pretty curious as to what these aliens are doing here.
Find lobsters and crabs can be quite territorial.
Yeah,
They're quite feisty.
the crab's upset that we're in his backyard.
Have you seen them? Have you seen more crabs?
It's actually in that spring, you know what I mean?
First, yeah.
That's right. There's a different one that's in the bottom, so. Alright, so you okay for us to, look at the bottom then?
Yep.
Kind of a tight fit in the quiver that.
If it's not going to fit in the quiver, then I think one of the bio boxes we can drop it in.
Ideally not one with the coral colonies, please.
Okay.
Corals are in, rock box A, right?
Yep.
Yes.
I show only one thing in B.
Yeah.
With that, if it needs to.
Yeah, we can put it in B if we need to.
I think so.
It's bigger than I thought it was now that we see it next to this. Oh, there's something inside of it.
Yeah, that's what we really think. Yeah, I think we should.
All
go
right.
ahead and just go for the firebox if that's okay.
Yeah, sounds good.
But if you end up trying to smash it in there. Want a picture of it? Drop it in or not? Thank you.
Very nice.
Something in it, isn't it?
What's that?
Is it possible to get the lasers on it somehow?
Yeah, hold on.
There's one right below it. Might not be big enough. There's the other one. Ooh, it's big. Wow. Is it a shrimp that's inside of it?
Likely.
It
That's
rem-
perfect. I got a photo of the lasers.
Okay. Good. Yep. Okay, so wire box 1B.
1B, please. So BioBox 1B.
Yeah.
I can pour it on in the wet.
This reminds me of Horton Here's a Who with all the small world inside of the sponge. is going.
Sebastian, control room. We have not turned off the clear.
Zoom in.
Okay.
No, I'm not.
Bridge clear comms might not be working right now.
Oh,
So fine. I think barely can fit us in here.
Yeah. We're gonna need a bigger box. Looks like cauliflower. There we go! Tucked in. Before we pick up, can we get a rock with no bio on it? I'm not logging bio. It's bio 25 in rock box 1B. So if
We're gonna get a rock.
yeah, if we can get a rock with no bio on it, and then see if we can then get a rock with maybe a yellow sponge on it.
2 rocks.
2 rocks. 1 bio rock, 1 no bio rock.
Not yet. Control.
And any of the rocks are good, so.
You can do a little poking.
Yeah, poke around.
Not that one.
Yeah.
The sharks keep pushing back.
Yeah. I don't want this. Nothing less than below it.
Okay.
The one below it, right below your arm.
That's probably gonna fall. Let me know, poking. No, just in case. Can you look up a little bit, like kind of look in the middle? A little more zoom.
Now we're gonna try to get rock.
About.
So if anyone watching is wondering, we are sitting on a ship, the brand new RV Falkor 2 in the mid-Atlantic Ridge. We are about 23 degrees north latitude. We're just off axis on the ridge, currently at a water depth of 1100 meters below sea level. And we have this remotely operated vehicle that we have a whole team operating. We have pilots and people who operate these manipulator arms to collect the samples, and they're doing that from the ship, and the ROV is connected with a tether. And this ROV's name is SuBastian. So the ship and the ROV are all operated by the Schmidt Ocean Institute, who has given us the time and access to these tools so that we can collect samples and other kinds of data and do some really cool science. All right. So that's really small.
You don't want it?
It's a little bit too small. Woo! By rock, sorry.
Oh, that might be a little one. Decent size right there. Maybe.
We want it to be like at least one fist size, if not two fists. Yeah, that looks great.
ROV breach, come check.
Loud and clear.
Loud and clear. Thank you.
Here.
Ooh, yes. Nice one.
Alright, this guy has bio on it.
It's okay.
Where do you want him?
It's going to be rock box 1C,
1C.
and I just need a couple of photos of it before it goes in.
No problem. Okay, over here.
That's perfect. Yeah, one moment. And 180 degrees on the other side, please. Perfect. All right. Instill that.
Then across the way.
Oh, the other one. Yeah, sorry. The
No, that's, yeah, you want one.
one is on the right and two is on the left.
Gotcha.
Perfect. Thank you.
You said you want another one, right?
Yeah. So, did you still want a rock with some of the yellow sponge on it?
Yeah, that would be great.
All right. Yellow sponge rock.
And that would be Geo 28.
That would be Bio 28-rock.
Yellow sponge rock.
Yellow sponge rock.
Which one do you want to come with? Thinking maybe jumble it looking ones here.
We don't want it to be dead sponge though, so it has to be the bright yellow one. So maybe in the just up a little bit.
Sure.
If there's
Just that much in shadow, but I could go here. Just up this way.
Yeah, I don't know if something in there can be grabbed, like that one in the middle right there.
Yeah, no, I want. There you got them.
Mm-hmm. It's a good looking one. It fits. If you can get the claw in there.
Too small.
Yeah, not enough bio on there, I think. Let's see if that rock is altered.
That's better than shadows.
Ooh, we? Oh, almost. It's like the claw game. Woohoo! So that will be BioBox RB1D. So on the far right on the bottom.
You want a picture of this one too?
Uh-oh. Is that still enough sponge left on it? It kind of fell off. I don't need a picture of that one, but it's okay.
And this is gonna go in BioBox 1D?
1D.
1D.
The bottom right.
Okay, I'm gonna move out of the way, just do the lid.
Could we grab one picture?
Oh,
You meant the crate 1D,
yeah,
right?
1D.
1D.
Rockbox
yeah.
1D, yeah. They do want a picture before it goes in.
Okay. Just there, I'll do that.
Okay, we're good, thank you.
Right, we're almost at the top of the summit.
Is that 1D?
1D, yes. Excellent.
Just not sure if I hit these quarters too. Right there. Yeah. So, right and right.
We'll picture.
Alright. Good.
To the summit. We are at 1100 meters. The summit is at 1075, so we've only got another 25 meters to go to get to the summit. We'll see if we see any coherent lava flows between here and there, but I don't think we're gonna stop for any more sampling. But I always say that, and then I always see a really cool rock that I want to grab, so don't hold me to that. Let's summit this mountain. Also looking to see if near the top there's any signs of hydrothermal activity. So that's hot water, hot seawater leaking out of the rocks. We don't have much to go on besides a cruise in the early 90s that they described one area with that, but we'll see.
Crab buddy.
It's a little shrimpy. Oh, there's the crab, I see it now. So the crabs prefer being close to the summit while the sponges like these vertical walls. We're about 25 meters from the summit if you want to do another one before then.
Yeah.
Larger talus and more
there's more sponges around here. Not seeing any in situ flows.
See through it. See that? You can see through it.
We might be getting into some in situ stuff. The round lobate shape.
Sorry. There's you know, we've already passed it, don't worry about it. But there was a sea spider, maybe possible anthropod that I thought would be big to see one, but
Yeah.
That orange red thing?
yeah.
You don't wanna go back?
If it's possible, that would be great. If she's okay with it, if not,
Yeah, we can do that. We're almost at the summit, and then we'll have like an hour to explore the summit.
Sea spiders are pretty cool.
So we are just backing up a little bit because we went over a sea spider potentially. Oh, I see a shrimp.
is it a shrimp?
Yeah.
Oh, that's
Oh.
unfortunate. Okay. Sorry.
All right, we continue on.
I started on legs.
What do you think?
I saw it like that.
I told you that the guys are free.
Because we're going so fast, yeah.
You might just slow down.
No, no, going. This is a good speed. I'm happy.
I'm sorry.
My computer is 1 hour.
Wow, that's a lot of sponges. Yeah, as soon as we get into the coherent stuff, the sponges are super happy. I guess because those rocks aren't moving. So here we have some lava flows. If you can see the difference between the blocky areas and the flows. Let me know when you're firing another one. Oh, that's a nice pillow that's kind of deflated on the
Just
side.
started one.
You just started one, okay.
Piston 6.
Piston 6. So your sample number is HFS 29. P6. Ooh, I like the. Ooh. Nice flows. There's some big lobate flows. Or pillow flows. Depending on how you want to define lobate or pillow. Oh, that's cool. There you see a cross section of what a pillow looks like. So the outside of it is still preserved there. We're almost at the depth of the summit according to my map, but my map doesn't have great resolution, so we should be transitioning into sediment again. again, we have this manganese oxide crust on the surface. A little bit more talus here too.
Samples down.
Okay. So the time is 5:49. And that was an unfiltered piston, correct?
Yes.
And P6 is what you said. Seawater. And the depth range?
1082 to 1070.
Maximum temperature?
5.8,
Average?
5.8,
And volume?
635 mils.
Good. Well done. Ooh, and this looks like maybe the very top where we transition to a very flat material, and it looks like there's still some pillow flows that are in situ. Interesting. Nice. We don't need to get exactly to that waypoint, but just kind of over in that area is fine. Yeah, then we can head to waypoint 3. Alright, is this just more crust, or is this a lava flow? It looks more like a lava flow. A very flat one. That's cool. Some pillows over there too. So some like gentle lobe flows that are sedimented. think we can fly pretty quickly to waypoint 1. What is that? Interesting. Looks like some.
Do we have left in the night?
It's 3 o'clock. We're coming up in an hour and a half. Yeah, I'd like to grab a piece at some point. It looks like all of the talus. It's either slightly brecciated flows or cemented talus. Really cool. Interesting. Oh, look at all that.
Can we do a zoom on this, please?
Yeah, absolutely. Let's do it. You guys happy with that?
Yeah, it's possible to focus a bit on the white part.
Like a super zoom?
Yeah.
Doesn't look like anybody's home anymore. Okay, you're good with the zoom then? Alright, let's keep going. Maybe grab a rock opportunistically around here. Yeah, there's more of these lobate flows underneath too. These rocks look like they have a little bit of iron oxide staining. A lot of the rocks we've seen here have a black stain on the outside that's manganese oxide. These look a bit more iron stained. Can we... It's very interesting. Just a little bit too far. So what is going on here is underneath all of this are a lot of different lava flows, and this is kind of the rubble at the top. There's also a kind of a, I think it's probably a lot of manganese oxide crust. So this whole area that we've been coming up has been alternating between lavas in situ and pieces of broken lava and a lot of sediment that's mostly biological in origin. And those green lasers are 10 centimeters apart when we have them on. And I see a comment, this stuff looks solid. Yeah, they're all pretty cemented. They're just starting to poke around and see if any of these are going to come loose. The old scientific poker rock. Is it this? No. What about that one in the back that's really sticking up? Yeah, that one there. That one might be. wonder what the white spots are on there. That biology white spots on the rock there? What's little hydroids are potential. I mean, yeah, we can't see it very well from here, but on that rock it could be hydroids, which are closely related to coral, right? Did I get that right? Yes, I got a biology fact right. For anyone tuning in, I'm a geologist from the University of Toronto. My name is Melissa Anderson, and we've been doing this transect to really look at rocks through time. So at the very bottom of this cliff, the rocks were very old and progressively getting younger as we get to the top, even though at the top they're still really old. So we're looking at how the chemistry of this crustal section has changed over time. We're going for it. We're going for it.
I have my doubts, but you never know.
I don't doubt you. Ooh, there we go. Yoink. Nice. Very good. We're gonna put this in a rock box with another rock. Let's do 1A, because there's only a small rock in there.
1A, okay.
Yeah, once we get some photos.
My sheet is right, we still have 2 empty as well.
No, they should all be full. But we can look down and double check. And then 180 degrees. Look at all the animals on it. Oh, it's cute. Patrick, is that you? We are, sorry, can you do it rotated again on the other side? I kind of missed my opportunity to take a photo because I was looking at the star. Yeah, right there is good. Yeah. Perfect, got it. Get him in, get him in. So this will be rock box 1A. Aww. I was hoping he'd come home with us.
It's kind of push me. Yeah. It's okay. If you feel like it's snagging though. Bring your camera in. Yeah. Okay, camera's all in.
Excellent. Thank you.
Alright, so you go to swap. You good to? Yeah, I got it. Good shadows that. You find the other shadow looks like it.
All right, we're almost at our first waypoint that we have planned on.
Just gotta turn that off. You want to go right? I was gonna float up a little bit. Try to force my way so it's not crashing. Yeah, a little bit. What's that? A little bit lighter. You're like right.
I always have to drop them. Alright, so
Forge, I'm ready to do forge. I'm gonna float up. Did you have something else you want to grab?
not grab, but I'd like to do a close-up on that rock that looks kind of orangey ahead of us, just as we drive. And then pretty quick here we can go to waypoint 2.
This is the program that operates our such a set. Actuator 1016.
Yeah, so just a zoom of that guy right there on the side. I kind of want to see how much iron oxide staining there is in there. Yeah. Yeah, interesting. It's definitely talus that's been cemented together. Okay, we can go just a little bit further and then make a turn.
So when we start going south here, Chris, he's gonna keep under my starboard, are you? Or
Only keep them to the west of you.
yes, like they are now.
Yeah.
Okay. Because then they should be deeper that way, right?
Yeah.
As long as I stay on the high part. If I see something high coming up to my right, I'll follow that instead of the track.
This seems pretty flat here though. I feel like there's a bit of an optical illusion. I can't tell if we're going up a hill or not.
We're going uphill right now, but you're definitely right. It's a lot less.
So our depth isn't changing too much.
Way less than that.
I do want to get to the tallest point of the summit. Scale the mountain. So we will be coming off bottom. It's 3:12 a.m. here, and we'll be finishing at 4:30. So not too much time left in this dive. All right, let's swing around and start heading to waypoint 2.
Off today, check that for a short period of time. Sure. Chris, can you it's under lights, I think. Okay. All right, you're ready now, right? Yeah.
Oh, ooh, couple pillows. Oh, let me grab a photo there.
Sure.
Yeah, I think the whole substrate is lava flows, and there's some large blocks of oxidized stuff there.
There, you're off, sir.
There's some other shrimp hiding in the rock there. It's really oxidized. Very crusty.
Back on?
We just dropped out half the bank, stand by.
Oh, yeah, my camera keeps
Yep, you're not gonna be able to come back. We're gonna have to address this in a safe way to provide a bunch of stuff.
we're gonna have to reboot.
Yep.
Yeah,
I'm pretty sure. What do you have control over right now?
I can't really thrust around, so and my camera's sort of randomly going.
Alright,
We can end the dive here.
well, I mean, we just have to redo our telemetry. We have to do it either way. It's fine if we do it. It's just that power to
Yeah, so I gotta shut everything off then, okay?
Yeah, I'm gonna shut up everything before you do it.
All right, we're just rebooting some telemetry. We're gonna come back back after that's done, so if the camera gets dark, don't worry, we're still diving. There's a comment about having major dive withdrawals. I feel the same. During the pandemic, I didn't get to sea for years, and it's really exciting to be back here taking some really cool samples. I can answer questions while the ROV is doing some troubleshooting. You're not going to see anything on your screen right now because they're just rebooting. Question of what's in the box. We have lots of stuff in the box, lots of rocks and quite a bit of biology, some sponges and some corals, some different kinds of sponges. We went through a couple areas where there was just sponges as far as the eye could see on some of these vertical cliffs. We'll be right back after this quick commercial break. If anyone has questions for our biologists, now would be a good time and I can grab one of them. I'm super quiet, oh no. Can you hear me better now? We don't know how old this is, but old. Millions of years old. Whoop. There we go.
Sample done.
Okay. The time is 6:25. What was the depth range? I guess.
55 to 1060.
Maximum temperature?
5.8.
Average?
5.8.
Volume?
456 mils.
Perfect. Logged.
I'm just barely balanced on that one rock.
Actually, it's even at the bottom of that rock. There's little white stacks at the bottom of the
For white balance?
yeah.
There's a thing on the arm that you can zoom in on too that
not
might.
going to use that.
No?
It's not where you're actually going to be looking.
Oh.
It's in closer, so you're going to be going through more water. So it's
Ah.
only if you're in mid-water that you'll do it. Is that what y'all wanted right there?
Yeah.
Some cool little hydroids here. They're just adjusting the white balance now. Ooh, pretty. Look at the iron oxide.
All right, so if this is too unstable, I'll just move again for you, Chris. But if you can just, some of these look pretty loose, like that one.
Like that's the rock one.
Maybe you can just grab it. I just do a bulk of this. What's that? You okay with the Chris?
Yeah. All three of them.
We should have heading now?
Yeah.
Yeah, you're back on.
I don't do it. This oscillating a little bit.
Yeah, we'll see how this goes. I'm just balanced on one little thing, so I'm not turning. So turn the lowers on here, bring those shadows. Give that touch bit too. That way you can keep your dogs on. Those good happens. One of those.
Let's see, try to pick it up. It is crumbly, but if we're gentle, it might still want to come home with us. It's not coming? It's too bad.
Definitely looks like some loose ones over there. You can try and scoot over there.
Or even at the bottom right there, maybe? Do you see the sticking out one with the hydroids on it, maybe?
Kind of below the large one in the middle of the screen.
Yeah.
See what I'm talking about?
Apparently our audio has been super quiet this dive.
So you see the large rock right in the middle of the screen right under your arm? Yeah. See at the bottom of that, there's one that looks like it's sticking out a little bit.
Ooh, that one's also crumbly. Look how oxidized that is.
You wanted that cut in half, didn't you?
Can I just look at it first and then decide? Wow. That is old. That is like pure iron oxide. Sure, let's try to put both halves in.
And where do you want that?
2D.
2D. Alright, you okay if I move the porch out? We might have to move the starboard on it. Those hoses are kind of under it, you know what I mean?
I can get it right there.
You get it right there?
Yeah, put down your foot.
Sure.
And can I get a quick close-up before you drop it?
Yeah, the rock.
Yeah. That's good. Got it. I don't need you to turn it. That one's pretty clear.
2D, right?
Mm-hmm. Someone commented that it's like pulling a tooth.
Now you can go for this other one.
Yeah. Why not? It's very old rock looking. Yeah, that's okay. I'll just take a quick close-up photo right there. Okay, good.
Same place it's going?
Same place, please, yes. Rockbox 2D.
Okay, now?
Perfect.
Back in the port. No, fully retracted. No other samples here.
Yeah, that's good. I think we can just try to make it to the next waypoint.
Sure.
Unless something looks drastically different, I don't think we should stop. Let's see if we can cover the rest of this ground and see what it's like up here. Thank you very much.
Yeah.
There's a little shrimpy hiding in there.
Alright, can you take the starboard rail out?
With the starboard rail up.
Yeah, we grab the hoses.
And up a little bit with the science camera. Thank you, perfect.
Neat little fissure there, huh?
Yeah. It's a beautiful fissure. Oh, can we? Oh, perfect.
I'll get on one over here.
Alright,
Yeah.
pull it in now.
Inside really well.
It fell off the arm, so like yesterday, you can start rail and put it down for me.
Nice blocky lava flows on the left-hand side too. What we're heading south.
I want to look at the starboard arm for it. Okay, so then look again. Rail. Bring starboard rail.
All good?
Yeah.
Really cool.
Oh, that was a crack there.
Yeah.
Will you just follow on this for now?
Yeah, that would be great. Yeah, it's going the direction we want to go. What are those?
Those are squat lobsters, looks like.
What are those biology people?
Those on
The little dudes hanging out. Nice.
First try.
If you will you zoom in a little bit?
No, because then I might be wrong.
Some long pincers, huh?
Yes, thank you.
Tell you what, for long term too, it'd be nice if we could get series. Then you can start getting that muscle memory where you know what's where.
Yeah, it might be something.
I think we could just outdumb them on that one. We could just swap that.
That's cool. What's what? It's a fissure in the middle of the rock. Sponge heaven number 3. It's really an overhang almost now.
There must be a lot of current, I guess.
Yeah.
Rather large sponge.
Big guy. Elephant ear. Oh, cool. Yeah, it's the rubbly top of the lava flow.
Do you want to peek off to the right of this overhang a little bit?
Yeah, that sounds like a good idea.
Oh, there's a big something.
Something's there, yeah. What are we looking at, biologies?
I have no guesses on that one.
It's a coral?
I think it might be a ginger root.
Ginger root.
It is a coral, I think it's dead. I would say it's dead. That's correct. Dead.
DED. Carrying along. Very big sponge off to the left. What's that? Yeah, I think they marked the top with this little curly fry.
The curly fry, yeah. Coming over the edge of this thing, yeah, cool.
And this, I guess it's actually, there's another fissure here.
Yeah, I mean,
Pretty wild topography.
way more rugged than I expected.
I'm gonna bounce over here. Yeah.
Can we pick up a bit of speed? I want to see if we can get to this waypoint and get over the edge. It's right at the edge of another steep scarp.
Moving along.
Ooh, I want to get that photo.
Looking at?
Not need to look in there, I just want to note it, so that's good. Pretty deep scarp. Still going in the same south-southeast direction. Kind of irregular.
It's skipping up a little. Oh, I see, it's a pink dot, where they going? Yeah. Yeah, I'm looking towards the west of it.
I'm gonna take a sample here pretty soon. Okay, sample started.
Okay.
It's bag 21.
So your number is HFS-33-B21.
That's all.
Yeah, getting close to the end.
Well, we're going to 4:30, huh?
What's the maximum speed you got on this thing?
Top of Scarp is where you're.
Top of Scarp, yeah. We're gonna be going uphill to get there, I guess.
Not really a race car, but
Not a race car?
I will give it what I can give it.
We haven't installed any jet engines.
Not yet.
Okay, sample them.
6:52 is the end time that I have. And the depth,
1040 to 1050.
maximum temperature,
6.1,
average,
6.0,
volume.
451 mils.
Okay. Logged. So those are a lot of pillow flows that are exposed there.
Very early.
Our lead range platform is not really broken at the moment.
I hear you just fine, Eric. We're gonna be speeding up a little bit to try to get some distance, so if you wanna bring the ship down a little faster.
Our lead range, yeah, you have to call the range check down here and see if you can understand.
I don't know if anyone on the YouTube chat can hear me, but we're getting pretty close to the end. It's almost 4 o'clock. We'll be ending at 4:30 ship time, so another 35 minutes or so.
That waypoint before the end of the dive. So just want to let you know.
Bring the ship down and stuff.
Yeah. Okay. Thank you. Bye.
Ooh, some iron oxide staining there. Pillow flows. Oh, good, you can hear me again. So we're getting pretty close to the end of the dive. We're gonna pick up a bit of speed just to cover some more ground. We're seeing a lot of different lava flows. The lavas that we're seeing have a lot of kind of blocky jointing, but those are in situ flows. Some big fat sponges and a crab taking a munch there.
I think it's one of those now, the flows and make little edges, a little ridge.
So you can see in the wall that we're looking at here some of the rounded shapes. So those are some very old pillow flows. They now look pretty blocky.
I don't know what it is, but there's something weird about it. Oh. I don't know about you guys, but I definitely feel like I'm liking these beds. Yeah. I'm liking these beds now. Yeah, I gotta wait. Catch up a little.
So as we're moving with the ROV, the ship has to maintain a pretty close distance because our tether is only so long. So every so often we have to pause and make sure they're catching up and we're not getting too far ahead of them. And yeah, we're in a little bit of a flat area with lots of sediment right now. Should see some more blocky lavas ahead. Just have to wait for the ship. I think those little pockmark looking things are burrows from animals. The dark sediment on top is just pretty coarse grained and the light sediment on the bottom is finer grained, and basically all of this is biological debris. You can see a lot of these black spicules. We suctioned up some of that earlier so we could take a closer look at what's in all of this sediment. I mean, I'm wondering where it's all coming from. We're pretty much close to the top now, so there's not too much falling on top of us here. The current depth is 1,028 meters below sea level. We're coming up to the highest point of this massive. Oh, there's a little star, a coral, and a couple more stars hanging out. Whenever we zoom in on this dive, we see just an incredible amount of biology.
We're gonna make it here.
Oh yeah, we have half an hour. Alright, might be coming up to a bit of a slope now. Still more sediment.
What's that? Yep. All right.
Oh, little fish. And a shrimp, maybe? Or is that a crab? Oh. Can't see. It's a shrimp. Squat lobsters hanging out in the coral with this really beautiful, oh, this looks like a dike. Can we try to take a sample of that very dark rock? But first, can we zoom out?
Sure.
All right. Yeah, that's perfect. Do you think you could break off like maybe one of the pieces sticking up?
We'll definitely
Give her a try.
definitely give her the old color drop out.
Alright. I can ram out or take it like interfere with those hoses, Chris.
Hey, fine.
Fine. My starboard ram is so much faster than the port ram. Not sure why. Maybe some settings got reset or something. It doesn't look like it goes out as far either for some reason. I'm not sure why. You see that? Alright, anyway. Moving in. I'm going to try to not mess with these squat lobsters.
Look at that one on the left there. That's stick out like that.
What's that?
Look at that one that's on the top.
Yeah, okay. So maybe if I just park, I'm thinking I'll put both rams on the side over here. That's kind of square, and maybe you can reach it then. I don't know, because that one's quite big on the top. I'm not even sure you can get your hand right. What do you think?
Give it a shot.
Okay.
Yeah, it doesn't look like there's anywhere super easy to try to grab a sample, but
I'm gonna give her head.
again, we need like a rock saw installed on the roving.
Rock solid
Several different things on there to accomplish that, and
It's never worked.
it's done some things, but
Look at all the little hydroids, I'm guessing. Is that correct? Hydroids and squat lobsters and some coral. Yeah, I don't know if it's worth spending the time to take a sample, but if something breaks off quickly, then let's sample it. If it's being too difficult, then let's just move on. What's the red thing is a good question. It's unclear to me. I think that it could be something biological sitting on top of the rock, but it's very red in color. The rock itself has a lot of iron oxide staining too. Yeah, I don't know that this is gonna work.
Nothing else, we'll get some nice shots of squat lobsters. Coral.
Squat lobsters and black coral. Couple little sponges on the bottom.
Wait a minute.
The rocks are infected with hydroids. They have such long pincers.
Or maybe that little right there.
If that's not biological, I don't know. What is that? Is that a... There's a shrimp there too, and a tiny little... Is it a tiny crab? I can't tell. A shrimp. It's stuck. Alright, let's just keep going. The rocks might be a little bit. I don't think that's rock, but can we grab it anyway and just see what it looks like? Or zoom a lot on it instead? No, to the right, the bulbous thing sticking out. Up. Up, up, up. What is that? What am I looking at? Someone's saying Stoloniferans. I don't know if I'm pronouncing that right.
Do you want it?
No. Biology team?
And it looks super interesting.
They probably think it's stoloniferous coral, which is like.
That's what the internet says.
Yeah.
Do you want a piece though?
I don't need a piece. I can mark it down in our observations if you want.
Okay. Alright, no piece. Let's peace out. Saying goodbye to the lobsters.
Is that
Adorbs.
dwarfs?
They're tiny little.
I think I love them.
You think you love them?
Alright, you good, Chris?
Listen,
Yes.
what is that little colony on the rock face behind the cute crabs?
It looks like there's probably a crack in the rock that something has filled in. I don't know if that's.
There's a pink thing.
Can you point?
Yeah.
Do you want to zoom on it?
Yeah, just a quick one if that's okay.
Are you talking about the
What are we looking at?
We're right between the lights.
okay.
Between the lasers, there's a pink thing that we want to zoom in on.
left there?
Yeah.
So it looks like a little hermit crab.
Oh, that one's cute. Little hermit crab.
Yeah, cute. Okay.
I thought I saw a tiny crab.
That's what we got.
Is it a crab or a lobster?
It was a hermit crab.
Hermit crab.
Okay, everybody good?
We're great.
Carrying on, we're almost to the top.
We've almost summited this entire volcano. We've almost gone 1 kilometer vertical on this dive. Man, there's a lot of them. Look, it's like a whole family. That's pretty.
Sure.
The right point, make sure we're clear of that.
With that, with the
Let's
signs?
the one. Yeah. Alright, everyone. Ready to read it.
Everybody good? I'm not just leaving that one. Pretty much?
Yep.
Oh, that was going long.
Oh, that is long. Holy cow. You got your photo? Okay. Race to the top. We've only got another 20 minutes. Can we get to the top of this hill?
I take my sample at 7:20. Is that okay?
Yeah.
I would say course fast enough. Jump off that up.
Yeah,
If
we've.
you want to come up at 7:30.
Yeah, I mean, we only have another 20 minutes left, so whatever time you want.
Yeah, so I think about 7:20 is my last.
Okay. almost time for the dive to end. We're just trying to cover as much ground as we can in the last 20 minutes. Currently at a depth of 1,019 meters below seafloor. Seeing a lot more broken up talus now as we go up this slope.
Definitely a little sharper up there.
Some more coherent rocks. I don't. Ah, I see. Can you zoom out on the science camera?
I can't, but I can move it to put that in the middle of it.
Yeah, that would be great. There's pillow flows and a dike cutting across. That's perfect. Wow. What do you think the heading is? So we're heading south, southeast, and so it's southeast.
I can turn and get on one of them.
Yeah, can you? Ah, a bit southwesterly. That's good. Perfect.
Probably about like that. About like that.
Perfect. Can you turn back around and just look at it for a second for me?
All these black corals too. I don't think we saw those deeper, did we? And not the one that's like super duper protected and white. Yeah, but it makes you weed out. Yeah. You get weed out, get you take it.
Cool. So this is what we call a dike that is intruding into older flows that are really rubbly. What do you think the width of that is? Maybe less than a meter.
You see the 10 centimeter mark, right?
I don't on there.
So you see the two green lasers?
I can't see them. Oh, there they are. So it's
that's
50.
10 centimeters, so that's 1/10 of a meter.
So 50 centimeters maybe wide?
For the whole, oh, you mean like that direction?
Yeah, like width-wise.
Yeah.
Yeah, okay. Perfect. We can continue moving.
Turn it on.
All right.
In and out again.
Almost reached the summit.
In 10 meters, I think that's the shallowest we've been so far.
Oh, shallowest, we're coming up on 1,000 meters, so we're just about at the highest point here. All right, can we grab a rock?
Sure. Just anywhere here?
Yeah, preferably one of the ones that look darker color rather than reddish in color. So do you see the little pile there on the left? That could be a good spot. Yeah.
Well, they need to repave down there. Look at that.
Yeah.
Come back.
I'm starting my sample.
Okay. And what is it going to be?
Piston center.
Unfiltered piston?
Yes.
So it's HFS 34 P7.
Keeps it nice and smooth.
Whatever degree, so, which is good when you're doing 360s.
Gotta find room for this rock now.
Stable-ish. No, no. Go more forward. Yeah. It's interesting. It looks like you can see impressions of rocks that used to be in that. Can you see that? Just near that hose? Yes, it can.
Okay.
I'll show you on the side.
Cool. Oh yeah, they've just, it looks like they've been plucked out.
So they must have been here right before we got here.
Yeah.
Alright, so Weller's just looking for darker colored. See what we got around here. So like any of those kind of darker color ones right there? you okay?
Yeah, something fist size. So the triangular one or one of these little two guys on the left would be good.
One right above the other.
I think I'm gonna have to come off this ledge.
Yeah, just sit flat.
Just keeps spinning me. What was that?
Then we'll let the red one, alright?
Yeah, perfect.
Which one are you doing, port arm or starboard arm? Probably starboard arm is better for the unit. Move over so much.
Sample stop.
Okay. In time 7:21. And I guess the depth didn't change much. What did you get for the depth? 1006.
Oh yeah, 1005 to 1006.
Temperature max.
6.4.
Average.
6.2.
Volume.
685 mils.
Perfect.
Definitely something interesting going on. It's making all this stuff.
Yeah, I think that it's actually right on top of lava flow with some manganese oxide, so they're all just cemented together. You can see some of the bigger blocks, like the one at the top of the screen, where they're really like glued together, the talus.
Is that one right there?
Fine, yeah. The one in the reddish one is fine too, just whatever is easy. Oh yeah, that's... Or the one right under the hand there, if you can, the dark one. If that wants to wiggle for us. No, he's stuck in there too. Alright, the reddish one that you were pointing at is okay too. None of these rocks want to come home with me.
That one I might be able to do.
It looks like they're just sitting there, but they're not. I guess this will be the last rock I'm allowed to pick up before we end the dive. Alright, let's take bets. Is it gonna come up? Oh, it's stuck pretty good. It's trying. Oh, yeah!
You can see that the crust that's glued to the surface.
Yeah. I just want to take a good photo. This is going in. What did I write down? 35 rock box 2C, please. I do, but let me do this first.
You want a 180 as well?
Yes, pretty please. Can I get a 180?
You ready now?
Yep. Pretty rounded. Lots of iron oxide and manganese oxide. This is a very old looking rock. Alright. What did I say again? Rock box 2C.
Okay. Alright, I'm bringing my camera in. I'm going to see about pushing out. I'm not sure if it'll rub on the ground. We'll see. Here goes the torch. Getting stuck on the jaws, on the claw cutters.
Alright, can you do what that does?
Zooming out.
All right, that was our sample 35, and I think we are done here. We have 3 minutes left. If anyone wants to zoom on anything, biology team, do you want to zoom on stuff? I'm sorry? Yeah. We have 3 minutes before 4:30.
If we could get a zoom on those sponges with what people consider the stolen corals, it might be nice.
Oh, sure. The ones right to the left of here. These guys?
Yeah, any of those sponges really. Like the big, yeah.
Or you want to go look
The
at
big
one?
fat one?
Yeah, the big fat guys.
Well, there's another little hermit crab.
That's
Whoa.
in the cracks there?
Yeah, gooey.
Kind of weird, huh? All right.
Ooh.
So I'm going to come up and go over to that big sponge.
You can just zoom from here. You don't need to move.
Got to move in, right? I'm sure you want to get a better view.
Okay.
We're considering this the top of the scarp, yeah.
Yeah, this is very good. There's a shrimp hiding over there.
Yeah, there's a little community I don't want to mess with over there, so I'm going to go around.
We can just sit and look at them.
There's the squat lobsters there.
There's a shrimp hiding in that crack.
Look at these weird cracks over here, too. So,
Yeah. It is like a cracked pavement.
yeah. Alright, so, I think you guys wanted to see the one over.
Oh my goodness, those are massive.
That's big.
Oh, you really saw some big ones. I didn't even notice
Well,
those.
you said the fat guy, so I'm taking the
Fat guy. We're gonna see the fat guy.
Lady, I know fat guys.
Is this making anyone think of like mushrooms after watching The Last of Us? I just keep thinking
Yeah.
of the flangey things and going, it's cordyceps.
It does kind of look like those ones, I forget what they're called, but they grow out the side of
Yeah,
logs.
the
Turkey tail,
flat
is it?
ones.
I think it's turkey tail.
Turkey tail. Yummy.
That's by a weird rock, too.
Okay, quick zoom, and then we are done. There's some little dudes living on it. What are, oh, they're tiny little lobsters, and so one little a couple little lobsters and a couple little
Oh,
stars.
yeah, a little spot lobster.
Do you see them? Are they babies?
Can you move that laser just right onto that sponge?
Sure.
Thank you. Perfect. Awesome.
I feel like we should move that laser where it's more in the center of that camera, too, you know?
So it looks like whatever these are.
Someone called it a polypore online.
Do you want me to try and sit here and get a really, really, really super zoom, or?
Maybe on just like those individual polyp things coming out, like the.
Like on the bottom there?
Let me get settled here a little.
So for anyone who didn't catch the whole dive, there was some really exciting stuff after, I don't know, maybe hour 3, where we hit a vertical rock face with just incredible numbers of sponges and other creatures as far as the eye can see. So the whole dive will be available after we're done. So after the live stream is done, you can go back and fast forward to those exciting parts. So we're just gonna zoom in on these little polyps.
Yeah, if you could zoom like up here where you can count the tentacles.
At the top, she's our biologist Monica is going to count tentacles on polyps. Ooh, look how zoomy we can get.
Yeah. Okay. So maybe the people online could give some insight, but I thought stoloniferous corals, I thought had 8 tentacles, and this has much more than 8. So correct me if I'm wrong. If I can gain some knowledge here.
Yeah, anybody online have a guess of the identity of this sponge? It's coral, I guess, not a sponge.
Well, this is so all of these little. I'm sorry, but
Come talk to the thing. Our biologist is going to quickly make a comment.
Okay, so these little polyps that we see on the surface of this sponge. So the big amphitheater structure is a sponge.
It's a sponge.
But these little polyps that we're seeing are everywhere. They're on the rock as well, beneath, like where the sponge is sitting on. You can see them dotting all of those rocks.
Oh yeah.
So what the readers, the watchers were saying was that these are stoloniferous corals, which are corals that grow on other things, essentially. And so the stoloniferous corals that I've seen have 8 tentacles, but of course these
Someone's
have
saying
much more than this.
Someone's saying zoanthid.
Okay, yeah, maybe a zoanthid. It's true. Okay. All right.
You got a picture of that shot?
I really want to Okay, now I do.
All right. we can take off. We're done. Well, thanks for tuning in from the middle of the Atlantic Ocean. This is the Schmidt Ocean Institute's ship, the ROV Falkor 2, running the remotely operated vehicle SuBastian. We've been taking a lot of rock samples and some bio samples in a very old part of the seafloor. Normally we're diving in areas where it's very young, but we're in a very old area. And we've gone 1,000 meters from the bottom to the top of a cliff face. So basically from very old rocks to somewhat less old rocks. So our dive is going to be finishing up right now. So I hope everyone enjoyed tuning in to the bottom of the seafloor. And hope everyone has a great day or night, wherever you are. Over and out. Right? That's a nice way to end a dive. It's also nice to have a bit of a shorter dive. 10 hours feels doable instead of 24.
Yeah, I I think we can start now if you want to. Ready? Yeah. Yeah, I don't hate the schedule. It's kind of good, you know?
Yeah.
Good morning, how are you?
Come on, get a little bit of flying, and then fix a little stuff.
I'm not bad around.
In the end of our shift.
We are coming off the bottom now and recovering.
I got a lot of rocks, so I'm very happy. It's like, if we're diving too interesting in an area, then I don't have enough room for all my rocks. This one is a lot of rocks.
Well, I've been on rock hunting trips with my wife, where I'm pretty sure we exceeded the tolerance of the vehicle for that thing was like right low.
Yeah.
By the time we were done.
Oh yeah.
Have you ever seen Ohio Flint?
No.
So I'll show you a picture. Like